This is where KGDC becomes extremely important. KGDC is not a sequencing facility or a research laboratory. It is envisioned as a platform, a bridge between science, healthcare, agriculture, industry, and policy. I would like to invite my comparing partner, Kavita. She's a senior consultant at KDISC, to invite Dr. Unnikrishnan, Member Secretary KDISC.
Thank you, Dr. Raju. I kindly invite Dr. P.V. Unnikrishnan. Dr. P.V. Unnikrishnan is a seasoned Indian engineer, development practitioner, and a civil servant with over 37 years of wide-ranging experience spanning e-governance, decentralized planning, science, technology and innovation policy, and the transition to a knowledge-based economy in Kerala. As Member Secretary of the Kerala Development and Innovation Strategy Council, KDISC, and ex-officio Secretary to the Government of Kerala in the Planning and Economic Affairs Development and Innovation Department, he provides strategic leadership for statewide innovation, skills, employment, and inclusion initiatives, including flagship programs like the Young Innovators Program and the Kerala Knowledge Mission, as well extensive contributions to decentralized technology and development projects. He has also played key roles in advancing science, technology, innovation collaborations, digital platforms, and inclusive knowledge economy frameworks for socio-economic growth. I welcome you, sir.
Shri P. Rajiv, Honorable Minister of Industries, was supposed to attend this session, but unfortunately because of important political deliberations, I'm not quite sure whether he will be able to join the session or he might grace the session at some point of time. Distinguished scientists, clinicians, industry leaders, colleagues from the universities and research institutions, and dear friends, a warm welcome to all of you. We meet this morning for a special curtain raiser to the formal inauguration of the Kerala Genome Data Center by the Honorable Chief Minister this evening. This initiative places Kerala among the first states in India to think of genomics not in silos, but as a shared One Health platform connecting agriculture, animal husbandry, fisheries, and human health. It also looks at leveraging the biodiversity of the state, the very unique biodiversity of the state, to create what Dr. Raju briefly mentioned as the bioeconomy. The KGDC is conceived as a state-led infrastructure to store, analyze, and responsibly use genomic data so that our farmers, fishermen, clinicians, and the public health system can all benefit from the same knowledge base. From improving crop resilience and animal health to better diagnostics and preparedness for diseases like dengue and sickle cell anemia. This facility is designed to translate cutting-edge science into everyday solutions for the people of Kerala. We are already seeing the contours of these possibilities in agriculture and fisheries. Genomic tools can help us develop crop varieties suitable to Kerala's changing climate, strengthen biological control of pests and diseases, and support healthier, more productive breeds of livestock and fish. In human health, genome sequencing is opening pathways for more accurate diagnosis and personalized treatment, especially for conditions that disproportionately affect communities in regions like Wayanad and Attapadi. I would like to place on record our deep appreciation for Mr. Sam Santosh, governing body member of KDISC and a global entrepreneur in genomics, whose early vision and sustained support laid much of the intellectual and institutional foundation of the KGDC. He had consistently argued that Kerala's unique biodiversity and human capital must be pursued and leveraged through a genomic data bank like the UK Biobank, and that such a resource can anchor future advances in synthetic biology, biomanufacturing, and precision healthcare. Though he cannot be with us in person today, his ideas are very much present in this hall. It's also my privilege to personally acknowledge the contributions of the Honorable Industries Minister Shri P. Rajiv, who has helped us substantively in taking the mission of KGDC forward, in initiating the major initiative in QSAT in establishing the Center of Excellence in Synthetic Biology. His determined efforts had been central to bringing modern knowledge-intensive industries into Kerala and to systematically improve the state's business scientific capability into jobs, enterprises, and long-term economic value for Kerala. As we begin today's Genome Parliament, let us treat this as a space to imagine how the Kerala Genome Data Center can become a truly public asset, a platform where people's innovation, responsible data governance, and world-class science come together to build a more resilient, equitable, and knowledge-rich Kerala. Thank you all once again for joining us at this curtain raiser event and for your continued support to Kerala's knowledge and innovation missions. Thank you.
Thank you, Dr. P.V. Unikrishnan. We are starting with the set of panel discussions right now. And I have a very good panel today for the plant genomics. The topic is from seed to system, climate resilient crops, food security, and new biotechnology for better crop productivity. And we have Dr. G.M. Nair, who's moderating the session. And the panelists are Dr. Rajalakshmi from Chemin Sciences and Dr. Anish Nair from NIMS. So let me take the privilege to call upon the panelists and the moderator. And I just want to give a brief introduction about the panelists and the moderator. Dr. G.M. Nair is an eminent scientist and academician. He's currently the director for Kerala Academy of Sciences. He's a former director of the prestigious TBGRI and headed the department since its inception. He has many publications and talks across the globe, and he has been instrumental in various scientific initiatives in the state and is currently very active in the research and skilling ecosystem in science. Dr. Anish Nair is a distinguished molecular biologist and genomics expert, currently serving as the principal scientist and head of the Center of Genomic Medicine at NIMS, Medicity Trivandrum. With a PhD in molecular biology from University of Madras, along with advanced qualifications in bioinformatics and international business management, Anish brings over a decade of experience spanning molecular diagnostics, clinical genomics, medical epigenetics. Dr. M. Rajalakshmi is currently the R&D Director with the Global Research and Development for Chemin Aqua Sciences. For the past 12 years, she has led Chemin research groups on diverse projects catering to animal nutrition and health. She has numerous publications and patents associated with new products and processes in aquaculture and animal nutrition. Her bachelor's degree is from Kerala University of Fisheries and Ocean Sciences and holds a PhD in post-harvest technology from Central Institute of Fisheries Education. Now let's start with the panel discussion. Thank you.
A very good morning to everyone. I'm so happy to be with you to moderate this panel, which is very important in the context. Before I get into that, let me thank Dr. Raju and Dr. Amjesh and Dr. Unikrishnan and all those people who have been steering Kerala Genome Data Center, which is going to be launched officially today. I wish the effort of the government of Kerala and all those spearheading this activity all the best, and hope that this becomes an epicenter of genomic analysis, genomic dissemination of data, as well as implementation of genomic inferences in the improvement of plants, animals, and human as well. It's very important that we need to have discussion on genomics, because we know that we have a revolution happening these days. We have profound knowledge on the genomic architecture of most of the organisms. We have sequenced them, we have understood their functioning through the genomic aspect, and we also understand the interface between the different organisms. How is the diversity, biodiversity being managed, and how is the genomic diversity important in maintaining the life on Earth? Not only because when you speak of the human beings, human welfare is very important always. And for that, especially after we have started the One Health program, that the health of all the organisms in an ecosystem is very important. It's complementary to each other. So understanding the genomic programs which determine the functioning of many of the organisms. What are the interplay between the organisms by which the diversity is being maintained? As well, because my panel is going to discuss on the plant system, obviously understanding that genomics crosses all the boundaries. There is no plant genomics, there is no animal genomics, there is no microbial genomics. Everything blended together, because we know that the molecule that governs the genomics is the DNA, which is universal. Same in all the organisms. So we need to understand it, because we have mammoth genomic data available, we have databases available, we have functional genomic data available, we have transcriptomics, proteomics, and all this data, plenty around you, is loaded with a lot of information, but then codifying them, annotating them, and understanding and taking the best value out of it is very important. So for that matter, the panel discussion in the plant system, we have I thought maybe there will be many panelists in the group, but we have two eminent panelists with me. I'm sure they would definitely do that, because yesterday when this was assigned to me, that I was desperately trying to contact them. Both of them have different domains, not plants. So probably I had a hard time to convince, but both of them happily accepted Dr. Rajalashmi being an animal scientist, and Dr. Anish Nair, who is in the medical genomics. No connection with that of plants, but then yesterday it was difficult for me to coin the questions to them which would suit to them. And I have not gone for any technical questions to them, but so I'm sure they would be able to answer it properly. Now the topic, that is the plant genomics, genomics of plants, from seed to system, climate resilient crops, food security, and new biotechnology for better crop productivity. I'm sure everything under the sun is involved in this. Within about 40 minutes given to us for this panel discussion, I am sure we will not be able to discuss all these aspects. But whatever is possible, we will discuss it. But understand that whenever we focus, agriculture is very important, food production is important, food security is important, food value addition improve the food security is very important. And obviously, when you speak of agriculture today, it is at crossroad. Climate change is no longer a projected threat; it is a lived reality. The reason is that erratic rainfall. There's not much of erratic process happening because connected with the climate change. Salinity intrusion into the croplands, it becomes extremely difficult. So you need to have salinity resilient or salinity resilient crops or resistant crops to improve the productivity. Pest menace, because we have a lot of pest resurgence that is happening because the friendly pests are being destroyed by the pesticide, overuse of pesticides. Nutritional insecurity, and there are several other issues that is to be addressed. As I said, within the limited time, we will not be able to go forward. Plant genomics has moved from being an academic discipline to becoming a transformative tool for global survival. We have functional genomic data available for the microbes or organisms to be cultured that way. But when you speak of plants, animals, and other complicated organisms, functional genomics becomes very important.
From my heart.
Now, we have sequences available, wheat, maize, rice, and several umpteen number of crop plants. We have genetic modification and release of genome crops. So this is again a major breakthrough that has taken place in genomic revolution, because initially when the genome reading was done, we have gone for manipulating organisms, especially plants, crop plants, for improving the productivity. So we know that BT cotton, BT brinjal, GM mustard, golden rice, all these are examples where we have gone for genetic manipulations. Wherein you can move any gene because the versatility is available to you. Manipulatory capacities, technologies are available that you can borrow gene from anywhere and put it into plants, crop plants. Classical example is from Bacillus thuringiensis. We have borrowed the BT gene and put it into plants, made them resistant to that kind of pest, like cotton and brinjal. But understand that when we go in for manipulating that way, people have a feeling that when you borrow a gene from another source, that it may create problems. So there were a lot of mudslinging, except for BT cotton. No other crop, though we have number of crops available, number of organisms available where genetic modifications has been done properly. None of them have gone to the farmer's field, though they are in the pipelines, but then the technology is available, but I do not know, maybe that there are people who definitely go against genetic modifications. So when you look at the BT, because it has done a lot of help to the farmers, the BT cotton, the bollworm, the leptodactyl larvae, completely destroys the canopy, but then they say the one which is made resistant through the genetic modification resist it, but then only this crop has gone to the farmer's field, and I'm sure that farmers are benefited. BT brinjal is again another technology which is being, with a lot of inputs we have done it, but I'm sure it's not going that big way. Though we have, because there is a moratorium, because it's a food crop, that people are, have a lot of apprehensions on how we will tolerate the BT gene that goes into the donor. But there are clarifications being given, but that doesn't satisfy the legislators and as well as the policy makers. So that means it hasn't gone to the farmer's field. GM mustard is a very safe technology, that's because mustard is always a self-pollinated crops. To overcome that self-pollination and go for crossing to improve them, Dr. Deepak Pental from Delhi University has gone for this technology. where mustard was made male sterile through a technology which could be controlled specifically, effectively, and you would have high yield as well as crossing facilities available through this. But I tell you, 15 years, Dr. Deepak Pentel had to fight with authorities, but now I think there is controlled trial is being allowed, and again, I'm sure it would be difficult. Now, this is a very safe technology. I'm not going to the details of it because it's a two genes being put into, and then they play with each other so that the plant becomes male sterile. For making the pollination process, this is used, and later it revert back to normalcy. But I do not know that. Again, there is moratorium given to that. Golden rice, again, another one for fortified one, where vitamin A deficiency, because it causes a lot of problems, and as well as iron fortifications have been done on this. This technology is available. I do not know how far this has gone into the market, but then in other places, this is extensively being used. But then the novelty of this is that 3 genes are being borrowed to make a golden rice, where vitamin A, which we cannot produce, are being produced within the rice plants. That's why the name itself is golden rice. It would have solved a lot of problems. There's a lot of disturbance coming from the back. It's better that you please close that door, and if others who want to talk, please go. Serious people who want to have discussions, please be there in the lab. Discussions can be done elsewhere, please. Being an academician, I'm very particular about it. I'm sorry for that. Okay, so very important that, especially for the students, I'm just giving this. I know that my panelists are eagerly waiting to answer their questions, but understand that Genome-wide association, GWAS, enable precision trait identification, SNPs are being used. So you would be, now breeding has become very easy, especially genome-wide association of traits as well as marker-assisted breeding. These are all breakthroughs that is happening, and especially marker-assisted breeding, that you would be able to go in for finding out the trait through a marker and then go for specifically crossing them and following that marker in generations. Not even you'll have to wait for the plant to produce. Before that, you would be able to identify it. And that's also being done. CRISPR-Cas9, this is something very great about it. Those people who are mudslinging on genetic modification, when one's own genome is being edited, modified for a betterment, I'm sure that there should not be. But then understand that we have released, India is so proud, the first genome edited rice varieties have been released by IAR, Indian Agricultural Research Institute, PUSA, and it was released in 2025, and maybe just see that associated with that, the NGOs have said that activists asked center to withdraw genome edited rice varieties. Maybe I said we don't understand the potential of this, Maybe that I agree that genetic engineering probably is something which is against nature that we are doing. But genome editing is correcting it. Human beings, of course, most of those genetic diseases could be edited, I'm sure, through genome editing process, that you could control them. So all these are available, but still people are against it, and they have gone to the court for stopping this. Transcriptomics, proteomics, metabolomics enable systems level proper understanding, because that this has taken up in a big way. Most of the schools are. Microbiome engineering for enhanced nutrient use efficiency and stress tolerance. I'm just introducing the subject so that it becomes easy for my panelists to answer, or the experts sitting in front of me would be able to answer. Microbiome engineering is something which, because you will be able to manipulate them for your, and especially as you know that when you speak of the gut microbiome, the role, the beneficial impact of gut microbiome on the human survival, human functioning, physiological, biochemical processes are all controlled by gut microbiome. I'm sure this is very important in this. Digital phenotyping, this is the most modern digital phenotyping and AI-driven genomic prediction for climate-smart breeding. The predictive models, so you could ease your breeding programs, you can predict and you can exactly bring in, because once you have the AI coming into the rescue. I'm sure that this is a great area which need to be attended. So these advances are helping us to move from seed, the genetic blueprint, to system. That's what, from a seed to a system, an integrated agricultural agro-ecosystem model. This is what the talk of the day, that you have to have integration of everything together. So plant genomics today is not just about yield, it is about resilience. It is about drought-tolerant rice, heat-resilient wheat, salt-tolerant millets, nutrient-enriched crops, pest-resistant varieties, and sustainable production systems that reduce chemical inputs and enhancing productivity. This is what is needed in the agriculture sector. They reduce everything, make them resilient to most of those adversities that is happening, and especially during the climate change. I'm sure this is going to make a lot of impact. Importantly, I would emphasize that genomics-driven agriculture directly impacts nutrition security, micronutrient sufficiency, and long-term public health outcomes. Food security is fundamentally linked to genomic introduction. Now, this is what the introduction I just want to give you. Now, we will get back to the panelists, and I'm so happy that we have two wonderful people sitting in there as panelists. You know, they're all trained people, though they look young. They are seasoned and they have a lot of experience in it. Their CV has been read out, and I'm sure they have great things to speak about it. Dr. Rajalakshmi is the first panelist whom I am asking the questions. She is director of Chemin Sciences, and mainly on the animal nutrition and aqua culture and related aspects, but she never had any hesitation yesterday when I asked her to be panelist in plant genomics. So that's the kind of confidence one has that when he has to speak. So with that, I would first ask the first question to Dr. Rajalakshmi. I will come later to you. The first question is, from an industry standpoint, how do you see plant genomics accelerating the transition from laboratory discoveries to commercially viable, climate-resilient crop solutions? What are the key bottlenecks in translating genomic insights into scalable agricultural product? It's a lengthy question, but I'm sure that she has understood it. You can just fragment it and answer it specifically. Dr. Rajal.
Thank you, Dr. Nair, for the very nice introduction. So as he was rightly saying, I'm not a plant genomics expert. I primarily work on animal health and nutrition. But then I would say I'm a big beneficiary of plant genomics, and I work very closely with the group of scientists. Within Chemin, we have a specialty crop improvement group who do a lot of work on plant genomics, and I work very closely with them. So there are a lot of inputs that we receive from plants that go into many of our products in various fields of application. So there are, as you were rightly pointing out, there are a lot of benefits that come at the primary level when it comes to the use of genomics, like whether it is whole genome sequencing or pangenomics or metagenomics. We get to identify, understand varieties or microbial strains or pathways, functions. There's a lot of things that comes open up in front of us. But when it goes to scalability, that is where the issues would be there, definitely. And because scalability is the long-term process. I mean, the whole process of scaling itself can take really a long time. There is no high throughput phenotyping that is possible, and so we have within Chemin a lot of molecules, like we have molecules like lutein, astaxanthin developed in marigolds, for example, and the whole marigold breeding program, the agronomy, cropping systems, it's a very lengthy process, so we I got to see a very interesting biomolecule expressed there, and I'm now really looking forward to when I can have it, extracted and use it. But then these things really take time. It would be at least 2028 when I'll be able to get the first flowers to be tested and used in my field. So that scalability is the first thing, and there could be several things happening during that phase, like whether it is a plant variety, there could be the gene environment interactions which would really express it differently from what we expected to. There could also be a lot of geographical variation, something that we observe when we successfully test in, say, in Spain, and then we take it to China, it may work completely different, and then we don't get that expression. Or whether it is a microbial strain that we develop through the use of metagenomics and further gene editing or selection, whatever it is. But then the scalability again could be an issue, because during the process of scaling up, there could be a genetic drift that can happen, and then we get a completely different output and expression. And this we have observed quite a few times, especially if it's a modified bug, there is a very high chance of genetic drift during continuous passage. So these are some of the issues. Also, it could be like a microbe introduced into an ecosystem like soil may not behave the same way, then there it gets into a lot of community interactions, and which can cause a lot of variations. So these are some of the bottlenecks. Another most serious bottleneck will be the regulatory part of it, and I think you were also giving some inputs on that. So if it is an edited or modified organism or a plant, clonal line or something, it can really take a very long time to get it introduced into the commercial environment because of the regulatory constraints. And countries like US are more kind to technologies like CRISPR. Europe is not. Europe is still very stringent on that. And whatever it is, the modification is, it can really take a very, very long time, including toxicity studies, ecotoxicology evaluations, and productivity, proofing of productivity in various cropping environments, various geographical systems, and also the biodiversity impact studies that will also, and these are very expensive studies to be conducted, which will also take, these are also very lengthy studies that needs to be conducted. So these are some of the things that at this point of time, genomics is really, really helpful to get us to the target functions, molecules, expression systems, but then the rest of the process is still a lengthy process to follow, especially the regulatory environment. And finally, as you said, the adoption barriers, whether it is from government side or whether it is from the public side, there could be adoption barriers which we will have to overcome through more awareness, education. That would also be a requirement there.
You have partly answered the second question also. You want to precisely answer some of those integrating genomics with sustainable bio solutions. The question says is, with increasing focus on microbiome based products, biostimulants and precision agriculture, how can genomics guide next generation agriculture inputs that reduce chemical dependency while maintaining productivity and global food safety standards?
Yeah, of course. Genomics would be that would be the key contribution that would come from genomics. Like you were talking about the GWAS studies that could be conducted, which we would be able to select strains, or we would be able to identify certain traits. And if we are doing a complete, and that is, I think that's something that the initiative here can really lead to, because we really need to have a very nice population structure for that, not missing out some of the unusual varieties, wild varieties, which will really lead us to some interesting traits or interesting functions. and which are helpful. For example, disease resistance or salinity tolerance, or it could also be drought tolerant varieties, and naturally these ones will need less inputs, chemical inputs. The other aspect is the microbiome based products. So the soil microbiome is a big area, I think, and I also use that in the area of aquaculture, because we also have soil as the fundamental base for the cultivation. And the soil microbiome, like the shotgun sequencing kind of technologies, can really help us understand the microbial interactions, the whole microbial community, and thereby manipulate or modify that in order to develop systems which are self-sufficient, in terms of whether it is nutrient cycling or whether it is biostimulants or biofertilizers. All of these can actually be achieved through this. And so that would be another important benefit of using these technology. Thank you.
Thank you, Rajalakshmi, for that precise answers given, though the questions were very lengthy and, there were a lot of components to be answered, but I'm sure she has answered it very clearly and precisely. We'll get back to you later if some queries come to Dr. Anish Nair, a medical genomics person working in NIMS Medicity. I was reluctant to call him and include him because I know that medical genomics and plant genomics, how it can be connected. But as I said in the beginning, that it's all interconnected, that medical genomics and plant genomics, if plants are not there, if there is no human health, and if the microbes are not there, there's no. So interplay between all these organisms is very important. So I was very happy then when he readily agreed and said that he would answer. My first question to Dr. Anish is convergence of plant and human genomics. As head of genomic medicine, how do you see parallels between precision medicine and precision agriculture? Can insights from human genomics, such as systems modeling and personalized approaches, inform crop improvement strategies? My first question.
Thank you, sir. First of all, let me begin by thanking KDISC, PV Unikrishnan sir, and Dr. Raju here for giving this wonderful opportunity. To begin with, let me say that, as you mentioned in the beginning, so what sustains life is 4 letters, A, T, G, and C, which forms our DNA. And that's across plants, that's across humans, that's across animals, that's across viruses, bacteria, fungus, whatever you take it. So as a genomics person, as a individual who's been doing molecular biology and working with these 4 letters for the past 15 years, I really felt that there is no differentiation when it comes to application of the tools. If you ask me about plant genomics, I'll be using the same set of nucleic acid extraction, the same set of library preparations, the same set of sequencing machines across all these phyla. So ideally, technically, there is no difference between working on genomics, even if it is plant or humans. Coming to your wonderful question where we can draw parallels between precision medicine and precision agriculture. So I believe there's a majority of students here. So I would just introduce what's precision medicine and then precision agriculture and then draw the parallels between them. So precision, as you know, means personalized, customized or bespoke kind of practice. Now that can happen in every field, right from getting your dress stitched. So when you have a function, you actually go to you have 2 options for getting a dress for your function. Either you go to a mall, go to a retail section, check out which are the dresses available, select whatever you want and wear it and walk off. Second option, you get the material from your vendor, go to a tailor, sit with him and choose what exactly and how exactly you want your dress. That is bespoke, that is customized, that is tailored, that is precision. Same thing applies to your precision medicine. So we have moved from one size fits all to the concept of individualized medicine. Now there's a lot of reason for that and it comes from, most of you would have actually experienced that as well. The same drug which works for your friend might not work for you, even if it is the smallest problem of a headache. So the same concept has been amplified and now being used across major disciplines. Primarily, and the most biggest beneficiary are in the oncology section, where we look at precision medicine. I'll give you a simple example. If you look at leukemias, when you're looking at leukemias, and if you happen to come across an individual who has a BCR-ABL fusion, that's a chromosomal fusion called the Philadelphia chromosome. So if that is there, then automatically the first precision medicine which came into prominence was imatinib for that. Similarly, if you're looking at a lung cancer patient, and if you happen to see that they have this EFGR mutation for that matter, then you would be, you have targeted therapies for that. So you basically look at those signatures and then tailor it accordingly. It's like how a mother of an infant feeds the baby when the baby starts crying, understanding what is the difference in the cry when he's crying for sleep, when he's crying for hunger. Same way. So the same practice can be applied into precision agriculture, where we can look at the sequence, we can look at the genome of these plants, understand what are the mutations, as was mentioning about the Jiva studies and the SNPs, the single nucleotide polymorphisms. The same thing can be identified, and according to that, you can tailor make your solutions for your agriculture, and that has to be regional. Because I truly believe what we see in the UK, the US, the Singapore or Germany or whichever country you talk about, or even if you talk about North and South India, the regional differences has a huge impact. So if you have regional, regionalized, like something like the Kerala Genome Data Center. In fact, my friends were very happy when I said our state has a Kerala Genome Data Center, and they were very proud. Oh, okay, your state is highly in a progressive manner. Because it's very important to document our biodiversity. So we look at these plants, we'll understand the genome sequences, understand their resilience, their genomic resilience, where they are tolerant to stress, they are tolerant to abiotic stress, or if they require more amount of fertilizers or they require less amount of fertilizers. All that can be very critically understood and accordingly provided solutions with. Now, coming to the second part of your question, can insights from human genomics such as system modeling and personalized approaches inform crop improvement strategies? Yes, of course. So we are in an age of AI, right? So when we are talking about systems modeling and personalized approaches with respect to digital advances, right now we have even moved more forward than genomics. So it's no more only genomics, it's multi-omics, where we are looking at not only the DNA point of view, we are looking at it from the entire central dogma of biology point of view. You look at the proteomics, you look at the lipidomics, you look at the entire thing connected with microbiomics, and we are talking about pharmacomicrobiomics. So it's not looking at individualized things, it is looking at the whole picture for an individual. So the same thing can be applied for agriculture as well, where the phenomics, what you see phenotypically in the plants can be correlated with their genomic structure, and then personalized strategies can be applied into the field for the farmers and for the people involved and the major stakeholders in that. So yes, systems modeling and personalized approaches can also assist with crop improvement strategies. Sir.
Brilliant connect being established. I'm sure this is very, very difficult area to connect to the precision medicine, precision agriculture. You have wonderfully connected it. Thank you. And to a second question, with gene editing and genomic manipulation becoming more precise, how can institutions ensure responsible innovation, ethical governance, and public trust while advancing climate resilient biotechnology? This is a very important area which need to be addressed, because public awareness is very important. When you do all this great intervention in crop improvement, we have a responsibility to convince them too. So maybe that, how would you?
So I believe the first step to ensure responsible innovation, ethical governance and public trust is to involve everybody into it. It has to be a public engagement. It has to be an involvement of all the stakeholders. So if you do not connect with the farmers and it is just a few set of policy makers sitting and making policies, or a few set of scientists sitting in their lab and doing their research work and giving out a product which is not the actual requirement of the farmers, then there's no point in that. So if we have to be articulating about responsible innovation and ethical governance, I think the first initiative should be involving all the stakeholders, the public, the scientists, the farmers, the upcoming researchers. Everybody should be brought on the same platform, understand the problem statement from the public. What are they currently facing? So a farmer based in Kerala would be facing a different problem than a farmer based in Haryana maybe. So you can't apply the same policy and the same methodology in both the places. The climate is different, the people's food habits are different. There's so much of difference in all the platforms. So regionally, like for example, probably KGDC can take up this forward, where they can actually bring all the stakeholders, the local stakeholders, who can put in their point of view, and the scientists can tailor make the solutions according to that. So I think that would be the major step in ensuring responsible innovation, ethical governance, and public trust. Secondly, an awareness program where you involve everybody from school children to adults. So if it's taught in schools, or if there are programs being conducted in schools where they know what is a genetic modified crop, or when you're going for a plant which is more resilient and more adapted to its the actual surroundings, how is it benefiting us? And if they know that how the farmer is benefiting, and if they are shown the larger picture, I'm sure they will start imbibing from the beginning of their life. Same thing goes with adults. If they know how exactly it's being benefited, I'm sure there are many people today in Kerala who have shifted from agriculture to other professions, just because they think that agriculture is not profitable. Now you have a wonderful set of tools available, the most advanced tools available, which can make your farming much more profitable than what you get paid in a 9-to-5 job. So, and ultimately you'll be, where you are born and brought up, you are back in your land, you're utilizing your land, doing an ethical practice and making the most amount of profit rather than probably in a 9-to-5 job. So I think that's lack of awareness. So if we create awareness amongst everybody about how agriculture can actually be made profitable with the advancements, with in introducing genomics into the field of agriculture, I think that will be the way forward.
Thank you. The only another group of people who could be convinced of what is happening is the activists. You know that they are doing much of damage to the genetic modification or any kind of manipulation. So maybe that instead of, in addition to going to the students, general public and all, the activists are to be educated properly, because they won't mudsling on all these things happening. So probably that should be added up, otherwise brilliantly answered. Now I have questions. We should go to the public, and I want, the audience. There are experts sitting here. Rather than becoming a monotonous kind of, this question is being asked to the panelists, common question, or to the people sitting in front of me. In biotechnology research, how is plant genomics evolving from single gene trait discovery to integrated systems biology approaches, combining transcriptomics, metabolomics, phenomics, and AI-driven breeding platforms? Are there anyone who could answer this specifically? Dr. Ramchand? Sir, you can. In fact, I had designed this question for you. You opted out to become in animal science. So please give him a microphone, please.
Thank you. So you again caught me in the same thing. So whether it's plant genomics, as you said, we are moving from single gene trait to integrated systems. And thanks to AI, this is going to totally change the way we are going to look at it. It probably fastens the way we are going to look at the outcomes of the genomics. Either it could be transcriptomics or so systems biology in particular, with modeling and combining transcriptomics, metabolomics, and phenomics, is going to change not only plant-based systems, human angle also is there, where we will be able to identify and then, uh, pathways that are going to make a significant difference in both diagnosis as well as in therapy. And that thing, I think it's going to happen in agriculture as well. In fact, more faster than in a human system.
Thank you, sir. Anyone who wants to supplement this? Dr. Unisha? Dr. Valsama? Any one of you? Dr. Sugunan, you want to? Come, please give him the mic. You know, it should be participatory. It need not be a monotonous kind of thing. Let it be a dialogue. I know Raju will stop me whenever time.
Okay.
Okay, no problem.
Am I audible? Yeah, okay.
Please.
So, in this era, I think last week there was a New England Journal article which said that you can have a genome analyzed and sequenced within 4 hours. That's a record. So it's an expandome sequencing method. Roche is doing it, and the equipment cost only around 1 crore. For a state like Kerala, I think investing around 1 crore will have its own advantages, because once we get the biodiversity of the Western Ghats sequenced, it's data available to all scientists who can then contribute to having the interactive genes and see how they can be utilized in functional genomics. So I would say that the priority for the Genome Data Center would be to have all these biodiversified material sequenced and have a data center which is available to all the people who are interested in analyzing it. Okay.
Thank you.
Sir, if I may, here.
Please.
So this just reminded me suddenly of one of my conversations with a consultant at State Forest Research Institute in Tamil Nadu, Chennai. So one of the projects that they were working with is basically combining, I will not say the entire thing, but at least the transcriptomics part, where they were looking at, so they happened to see that there is a red tamarind variety and there is a brown tamarind variety. And when they did an analysis of phytopharmacological analysis, they were able to tell that there were a high amount of polyphenols in the red variety than the regular brown variety. So what they went on, and polyphenols are responsible for more amount of antioxidant activity, which all of us with our stressful life needs very badly. So when they looked at the transcriptomics angle, they did sequence it and they did go ahead with identifying the SNPs and everything. But what interested me was they identified the entire transcriptomic pathway of this particular anthocyanin synthesis pathway. And they found that if they were able to add an elicitor molecule, they could increase the if they could spray an elicitor molecule which would increase the activity or increase the pathway, modulate the pathway such that the anthocyanin production would increase. And this they refer to as elite clones of the tamarind variety. And this is what they wanted to give it to the farmers and increase their livelihoods, because a plant which has a better yield, better properties would get them more rates in the market. So this could probably be an example where they are integrating transcriptomics as well.
Yeah, when he was mentioning about that, I also, reminded of an example where, some cases the whole integrated approach, including transcriptomics and metabolomics, can help in elucidating pathways so that it can be modulated towards a higher expression of certain target biomolecules. So, but then sometimes some of the traits could be like polygenic traits, which can be which can really become more complex to get it integrated into a cropping system and get it successfully expressed in a farmer's field. But then this has been successfully done, for example, for lutein and from Kemin, actually there is the lutein, high lutein marigold is being grown in India by the farmers in India. We have huge areas where it is being farmed. And there are several different aspects that needs to be looked at in addition to the expressing the pigment, but the pigment, the petal size, the flowering pattern, the shoot, spread, and all of these things will come into. So the integrated approach definitely is used, and it is very helpful in that.
That's very important, because when you look at the metabolic pathways that is operational in the organisms, you can externally manipulate them. Probably if you said about the elicitor or a stimulant that is available, this happens normally in the environment, There are factors which controls the metabolism. So if some useful products could be enhanced using an elicitor or probably an external stimulus, I'm sure that would be something which you can look into, because rather than looking at the genetic manipulation always. So I think that's a great thing. 10 minutes? Okay. Now, it's again another question common. If you had to identify one transformative, this is the end of it, one transformative breakthrough, technological or policy driven, that will redefine plant genomics in the next decade, what would it be, and how will it shape global food security? It's a little very focused kind of question, that how would you, this is very important, because just one strategy that could be adopted which can improve the food security. Anyone else you want to? You can also answer.
So I think we are right now sitting in a event which is inaugurating the Kerala Genome Data Center. I think that could probably be a transformative breakthrough, policy driven, where we document the local biodiversity of our God's own country, and not only just documenting. I am a staunch believer that data just stored is underutilized. So we should be able to apply it. And this data, probably rather than calling it just the genome data, we can have a multi-omics data, understand the biodiversity of our region, promote these advanced techniques into the practice of agriculture, and probably help the farmers realize the fact that yes, there can be wonders possible out of agriculture. So I think we are currently sitting in a transformative breakthrough which no other state has ventured into.
Very.
I would agree with that. Actually, further to that, as you said, there could be a lot of data that is developed, and we have a very vast biodiversity, and I'm sure there would be definitely several interesting varieties of plants or biomolecules or pathways or functions that we may come up during this the whole analysis of the whole data. And what I would like to just add is to also involve industry, because there are several industries that could lead this to different directions, whether it could be on the human health or health of animals, or it's for food applications. So I think that kind of a collaborative effort would be very useful for this.
Very great. Okay. What is important is the databases are very important, See, you need to have data to be put into the databases. There should be common databases which should go into the public domain. Whether it is biodiversity database or genomic database, I'm sure that the Kerala Genome Data Center, the main program would be to coordinate all these things, get all those collated and put it into something, into a public domain, so that everyone can use. Every time, what happens is that you compartmentalize, you contain everything to yourself, you don't put it across. So I think this is one of the major lacuna. Even biodiversity, when I was in TBGR, I could make out people were not in a position to give data which could go into a common database. So unless you enrich the databases and then probably put all those things, whatever you generate sporadically at places, and each thing that you generate would have a value that will reinforce the databases that generate. I'm sure there is a great hope. I'm stopping it. I'll just conclude it. Artificial intelligence and machine learning platforms are transforming breeding itself. Genomic selection models can now predict performance before field trials are conducted. That's very important. Multiomics integration improves. This is what the essence of what is being discussed. Integration, farsightedly I have compiled it. But I know that this is what that has come in. So prediction accuracy for complex traits like drought tolerance and yield stability. Crop growth models are being merged with molecular data. All these put together, I am sure there is a transformation happening and everyone is aware of what is needed to be done for improving the food security, because that's what we look forward, that food security, quality food, safe food, which will not harm the human beings, not the junk foods, the other things. So maybe that any kind of improvement in terms of beta carotene or maybe iron fortification or anything that is improved, I'm sure definitely reflect in the health of the human being. So that's what is to be known. So the future of food security therefore lies not in discovering one magic gene, but in understanding biological networks, environmental interaction and management systems as an integrated whole. That's what is needed. So I'm sure that this panel discussion with my youngsters sitting here, very dynamic, very knowledgeable youngsters who have answered all my very lengthy questions very precisely, very beautifully. I take this opportunity to thank both of them, Dr. Rajalakshmi and Dr. Ranjish. They have, I'm sure, with the kind of intervention from the audience, my students sitting in front of me, I'm sure that now you have a future direction that AVEX have, because crop security, crop productivity are all, we had green revolution, but then we need to have a evergreen revolution which will not harm the environment, the diversity, because we rest on a diversity which is on a very delicate balancing. Each component is very important. So unless we understand it, and now we have the supreme understanding on the genomic level, so I am sure that we will be able to understand the diversity, the interaction in the environment, and the beneficial impact of the biodiversity in a great way. I take this opportunity to thank both my panelists for the excellent way they have given the answers, as well to my best wishes to the Kerala Genome Data Center, a brainchild of Kerala. Kerala government is visionary. We are new in many things, including the declaration of state microbe, the Bacillus subtilis, a wonder bug as a state microbe. I'm sure several new things are happening in the state, and I'm sure with that we will have a great future, comfortable future in the coming. And I wish Kerala Genome Database, Dr. Unnikrishnan, Dr. Raju Ray, Dr. Amjesh, and all those being involved in that very best, and hope this center comes out as the best center in Kerala. Thank you very much, and thank you all for your patient listening. My special thanks to you. I was watching her, the way she is depicting, and I'm sure she has been doing this exercise continuously. Congratulations. Thank you very much. Please give her a very big hand in the room. Thank you very much.
Yeah, thanks to the amazing panel and Dr. J.M. Nair. The next panel would be on animal genomics. So the themes are livestock, microbes, One Health and fisheries. And the panel is moderated by Dr. Ramchand. Thank you. Yeah. Dr. Ramchand is the president and CEO of Saction Life Sciences and Mac Genome Technologies, Cochin. With a career spanning over 25 years, he has established himself as a prominent figure in the biotech and pharmaceutical industry. He holds advisory roles in multiple companies including Genome Labs, MedGenome, Theragen Life Sciences, and Theragen Biologics. I welcome Dr. Ramchand to moderate this session. On the panel, we have Dr. Valsamma Joseph. She is currently serving as the Director of National Center for Aquatic Health and the Course Coordinator of MTech, Marine Biotechnology Program. She was awarded PhD in Environmental Sciences on the thesis Aged Algae in Industrial Effluent Treatment from CUSAT. Her research interests are in marine algal biotech for various applications, including biofuel, animal nutrition, and control of microbial pathogens. I also want to invite Dr. Ananda Subramanian Muthuswamy, who is a professor of biotech at PSG College of Technology, Coimbatore, where he has been a faculty since 2000. He also has a PhD from University of Madras and has extensive research profile with publications spanning journal articles, books, and conference proceedings. With hundreds of citations and significant co-author network, Dr. Muthuswamy's work reflects a strong record of scholarly impact in science. I welcome Dr. Muthuswamy, Dr. Ramchand, and Dr. Valsamma Joseph to the dais.
But he can talk on animal genomes and only later he will come.
Dr. Ramprasad, do you want to join the panel too?
Yeah, I think you can come because because you are also there in the next session. So thank you, Dr. Ramprasad.
So Dr. Ramprasad is currently the CEO of MedGenome. MedGenome is India's biggest lab in genomics. So I'll invite him to the panel too.
Yeah, thank you very much. Thank you, Dr. Raju. Thank you, Dr. Nikishan. And we have some eminent panelists are here. One is Dr. Ramprasad. He is the CEO of MedGenome. MedGenome is one of the most successful biotech company and initiated with next generation sequencing about 10-12 years back. I was also part of that company when it started Genome. I was also in the advisory. We also have Dr. Ananda Subramanian, who is a professor dean at the PSG College. And we have also Professor Valsamma, who is an expert in fisheries. Now, in the previous speaker already mentioned that, the people who were talking earlier, they were not in that particular area, especially Dr. Rajalakshmi. I know that she was more of in agri-foods or fisheries, and so is the other people also. The same is the case. I also know I'm an organic chemist. May I request to close that door because, there is a lot of sound is coming, please. Thank you. Thank you very much. So I am an organic chemist and I have not worked with anything connected to animal genomics at all. But then, as you know, that ATGC is there everywhere, whether it is animal, whether it is plants, whether it is human, microbiome. So it doesn't matter. And it is also very easy to make questions and ask that it is their duty to answer these questions. Anyway, now the first question comes. Does Kerala need an exclusive genomics or, next generation sequencing of all that, whether it is human, because you know that, the biodiversity is extremely high. People have been migrating from several hundred or several thousand years. So is the case with animals, various type of animals, especially livestock. And so is plants. Probably unique plants can be seen in Kerala, especially with medicinal properties and microbiome. And many, many diseases are coming out very regularly. That is because of, varieties of biodiverse microorganisms are also present. Now, the question what we have to answer is that, whether we need a Kerala-specific genome data center, especially dealing with all the data of Kerala genome. That is whether human, animal, plant, or microbiome. And that is the answer that, in these panel discussions we are going to come out with. Now, probably Dr. Ramprasad will be telling that, they are already MedGenome has got Asia specific. That is 28 countries. There is Genome 100K, isn't it? Genome 100K is there that they have developed. Probably he will be mentioning about that. And why the same way, whether Kerala specific genome is required. So is the case with animals also, plants also, microorganisms also. Now, if you see, whether it is connected to animal genome, plant genome, microbial genome, these are all interconnected. Now, as far as human genome is concerned, why we are sequencing this? Mainly for the purpose of diagnostics or for pharmacogenomics. That means as precision medicine or personal medicine. And we also have a company called, it's on nutrigenomics called Well Genome. Mainly we are prescribing the supplements, nutrition, food based on the genomics. The same way, in other cases also, the base genome data is required or not, that is what we will have to answer. Now, as far as the humans are concerned, we know why we are sequencing this. As I mentioned that, because diagnosis has become easy, then precision medicine, personal medicine, this is the major reason for sequencing. And why we should this for animals. So I will tell you some example. As far as animals or livestock is concerned, you have cattle, you have animals like buffalo or cows. You also have other animals, mainly layer or broiler, and so various types of animals are there, and which is a part of the agri-foods. Very interesting example I will tell you. Currently, every day 300 million chickens are killed in the whole world. We are about 8 billion people, 300 million. Now, if you are talking about 50 to 60 years back, this was probably about, 30, 40 million. Now, during that time, it was taking about, anywhere between 6 months to 1 year to grow a chicken to become 2.5 to 3.5 kg. And anybody who is involved in this layer or broiler industry will know it takes only 35 days, 5 weeks, and the chicken will become, or the broiler will become about 2.5 kg to 3.5 kg. How this happened? This is done through selection procedure all over the world. Now, the same way about the layer, which is egg, producing that earlier only 30 to 40 eggs used to produce about 40, 50 years back. Now it is anywhere between 250 to 320. That's why the egg price is compared to 10, 20 years back, it is not going up. It is kind of maintaining. So now all these things, what I was mentioning, now just see about the cattle. India is the highest milk producing country in the world. We have overtaken the USA also. How this has happened? So we hardly had milk probably 40, 50 years. Again, this is by selection procedure. Now you think about green revolution, what has happened? When high growing plants were made by, Professor M.S. Swaminathan, and that was again by selection procedures. The question is that now from the selection procedure, can we go the real genomics way for developing all this, and that is what is happening. So I'm not going to spend too much time on this one. So probably my questions will be also related to not only animal genomics, little bit it will be human, it will be also with microbiome as well as with the plants also. My first question is to Professor Ananda Subramaniam. So, there is something called antibiotic resistance, which is coming up, and because rampant use of antibiotics. Another thing is that in broiler and layer chicken, there is something called antibiotics growth factor. Probably the food given is only 20 rupees per kg, and probably in about 35 days, about 10-15 kg food will be given, and there is something called FCR, feed conversion ratio, that should be really something like, ideally it should be 1, it is not possible. Now, in order to improve the efficacy of this feed, there is something called antibiotics are added, normal antibiotics, and it's a very small concentration. No one knows what exactly is the mechanism. Some people are telling it is to kill pathogenic organism, but that is not the thing, because the FCR improves significantly. And now this is started, like, most of the countries are banning this, using antibiotics. Now, one of the very important work Professor Ananda Subramaniam has done is about this antibiotic resistance, especially gut microbiome, and the transformation of one microbial to another microbial, especially plasmid transformation in the gut of many of this, whether it is for broiler or layer or other animals. So my question is on that. Can you please tell about your study, the importance of that study, and what is the future?
Yeah. Thank you, Dr. Ramchand. And I also take this opportunity to thank Dr. Raju for being here to be a part of this program. First of all, kudos to Kerala government for bringing out this progressive thought of having a data center on genome. This is going to really be a transforming path in the scientific field. Coming to what Dr. Ramchand was asking me about the work on poultry. So poultry is a very big industry, and we frequently discuss with poultry companies, and there is always a saying that they don't use antibiotics. So we wanted to look at how this antibiotic presence is there. So we started working on the samples of gut microbiome to look at what kind of resistance genes are there. And not only from our region, across the globe, looking at the databases available. And we found some interesting results. These results include a wide variety of drug resistance. Almost 218 variants of drug resistance were observed across the globe. And in that, if you look at it, mostly are, of course, these are things which are mostly known, that it is mostly transferred through horizontal gene transfer through plasmids. And even within that plasmids, you have two types, either mobilizable or conjugative plasmids. Of course, 50% of them are going to be a non-mobilizable plasmid, which means that they will be present in that particular region, but they will not be able to get transferred. It will be probably doing within and this mobilizable transmit can transfer within that own organism, whereas a conjugative one can move from one organism to another organism, which makes it more dangerous. In that sense, it's going to spread the disease very fast. So looking at that, it's almost 30% in our study that we observed that it is due to the conjugative transposon these antibiotic drugs are there. Luckily, if you look at it, we found mostly beta-lactam resistance genes that are going to be the most one. Of course, the last 2 decades we've been using that. We have not come across the recent one like colistin or vancomycin resistance. So that so far it's fine. But it also says that these gut microbiome has these type of, even lactobacillus, all kind of firmicutes and bacteroides have these type of drug resistance. It only says that they get exposed to this type of drugs. We cannot directly connect it whether it is being used in the feed company, but it says that there is a presence of these type of genes in this. And we also looked at it, it's not only the plasmid that is going to play a role, it's also going to be a phage, which is going to be a significant one. And there is a big difference between a European sample and an Asian sample. Almost 6-fold rise of beta-lactam resistance was observed in Asian samples, particularly China, and we even did some with our own samples. It was very similar. Our fold rise of beta-lactam is very high compared to Europe. So this also gives a clear indication of how drug resistance is increasing in this Asian region. In addition to plasmids, it's not only plasmids and transposon, there are phages which are going to play a very vital role in this screen. Almost 2 to 6% of the population of the data we analyzed showed drug resistance through phages. And you back again, you look at two type of phages. One is a temperate phage, another one is a lytic phage. And temperate phages carries the most of the drug resistance, better than lytic phages, which accounts for some 30%. It's also is going to be spreader of these drug resistance. So what we have done is actually we have now developed a tool to analyze the risk of given a metagenomic sample. We will be able to analyze the risk that is involved in spreading this type of drug resistance.
Yeah, very quick question that, do you think that antibiotics will be stopped using in animals soon, or it will take some more time? Because some of the countries have started, especially like France, and many of the people are using certain probiotics. I myself was involved in developing PB6, which is being used. So what is your...
Yeah, it is there in practice. I mean, in paper it is there already. We are not supposed to use those kind of drug. But we don't know what is really happening there. But it slowly, as it has to improve. That's what I said. The recent drug resistance are not been seen, except for beta lactams mostly. Of course, tetracycline is very common because we have been using for almost 5 decades. Right.
Thank you. My next question is to Dr. Ramprasad. So one of the things, as mentioned earlier, that, Medgenome was involved in that 28 Asian countries based genome, and why this was done, and what is the importance? Because we can really relate that particular Medgenome work can be like Kerala genome also. That is first question. And second question is that apart from human, I know that, mainly for diagnostic purpose, pharmacogenomics, especially, I understand that US FDA has approved about 45 such, whether it is SNPs, mutations, or genome variations. And apart from that, many other things are also is doing. But my major question is that mainly connected to microbiome and virus and animal genomics and plant, what are the things Medgenome is doing, or for others?
Yeah, thanks, Dr. Ramchand, and really appreciate this is very interesting. Kerala gets KGDC. I don't think any other state has such an initiative focusing on genomics, even though there are a lot on biotechnology and biopharma. But this is, if it's executed, I think it will be a different trajectory how the state takes this forward. So, talking on some of the genome initiatives we have taken, apart from the regular commercial activities that we do, 2017-18, when we started doing more and more genomic testing, one thing which we found was there was no normative data in the population. So it is without a normative data, it's very difficult to differentiate between which genes, which variants cause disease and which do not. Roughly about 15 to 20% of the rare variants we were unable to decide. This is, I'm talking about 10 years back, 9 to 10 years back. So that's when we decided that, okay, let's create a normative database. So it's nothing to do with the medical genomics or any kind of phenotyping, nothing we done. So we decided India is a pretty diverse country, especially you take whole of South and Southeast Asia, including Indonesia and other, very diverse, one of the most diverse geographies in the world. So it varies. If you take in India itself, there is a ancestral North Indian, ancestral South Indian, Central Indian, Tibeto-Burman on the other side. So very diverse. There are regions. Last at least about 2,000 to 3,000 years, there has been a caste-based practice. There was not much of random mating. So a lot of founder effects created across the country. So we wanted to basically understand what our variants are they significantly different compared to other populations in the world? How many unique variants are there? Are they anyway associated with certain diseases, or how we respond to certain drugs or even tricks? So this is a very high level at the 2016-17 we have taken, and we sequenced, it's our own investment that we have put, it's a large initiative. We sequenced more than 2,000 whole genomes. Today, we published it, we use it today for our diagnostic purposes. It's a fantastic resource that helps us to differentiate between novel variants and India-specific variants. There are more than 4 million unique variants that we identified in the population. As we sequence more and more, you get unique ones. Now, for a 1.4 billion population, sequencing a couple of thousand or 3,000 or 4,000 is not enough. Our diversity is much larger. And there are more and more utility and applications that are coming as to why we need to do population level sequencing. Many other countries have done far, far better work than what we've been doing. I think these initiatives like KGDC plays a very significant role. If it is taken in a very systematic approach, planned well, involve experts from both government as well as private, and do in a systematic, data insights are very important. How we create the data infrastructure and utilize all the industries and ask the burning questions using the data that's available. So that's the small initiative that we have taken, and hopefully larger initiatives comes up in the public infrastructure and government initiatives that happen both at a state and central level.
Yeah, my second question was on that. Apart from that human genome, we're doing many other next generation sequencing. And what type of samples you are getting, and what is this normally people are doing?
We sequence lot, roughly about 30 to 40 terabases of data a month, which we're talking about 1 to 2 petabytes of data a year. We do solutions, we do service provider, we get a lot of plant genomes, we get probably about 3 to 4,000 microbiomes every month. As a service provider, we do. We get different types of requests. We do whole genome microbiomes, 16S, 18S, whole transcriptome. What we realized is microbiome is far complicated than just doing a whole genome, human genomes. Dynamics are different. Subtle changes a lot in microbiome, whether it's a gut, whether it's a skin microbiome. For example, we work with many of the consumer goods companies where we do microbiome for cosmetics, for detergents, for soaps, disinfectants, how it changes the microbiome. Microbiomes are very dynamic, and it's not easy to understand how they change based on the environment, based on drugs, based on what you eat, why dysbiosis come. We can clearly see a pattern of what we call diversity index, or there's a index called Shannon diversity index. When you sequence a microbiome and analyze it, for example, you take gut microbiome, there are different types of microbiome that will vary. If it's a dysbiosis, you clearly see one specific type of bacteria increasing significantly. You intervene them either through drugs or through nutrition, the complete, again, Shannon diversity index changes. So you can clearly see at a very macro level distinct, unique patterns that come in. But it's still not it's still not a very precise science. It's evolving. The methods, the analytical tools, even the sequencing methods that we do are not long read has come. It gives much better resolution, specificity compared to short read. But it's not as accurate as how we do today a human genome. But it's fast evolving. The implications are large on that part of it.
Yeah, a quick question that, all this Asian genome database and Indian genome database are publicly available, people can use it?
So there is a larger initiative government of India has done called as Genome India. They finished about 10,000 whole genomes. I'm not sure whether it is open. We tried to download, but I think still not open for public. There's some kind of a policy decision that's being taken. Genome Asia, whatever is published in the paper, that people can access it.
Yeah. Yeah, before going to the next question, still that, the voice is coming from that side. So those who are standing here and sitting here can feel that. I request you, either those who want to talk, please go outside and talk and come back, especially if you are taking tea or something. And, otherwise it has become very difficult to interact here. My next question is to Professor Valsamma. Yeah, the fish tilapia. What I understand is that, now everywhere it is culturing, and probably 30, 40, 50 years that, the growth was slow. It was not cultivated and it was very expensive. And this has been developed through selection procedure. Now everywhere it is there, comparatively cheap also. It's a good fish. Now, if we have to further improve, what type of technology you might use, especially genomics? That is my first question. And similar, what are the applications of genomics? For example, white spot syndrome is a viral disease, nothing is there. The similar way, many other diseases are there in the fish industry, especially aquaculture. So what all can be be done so that, this can be significantly reduced?
Yeah. Thank you, Dr. Ramchand, and I thank KDC and Dr. Raju for this opportunity. So I will answer to this question, complex question in an integrated way, because unlike terrestrial systems, aquatic systems are very unique because we have to consider about the health of these animals, and we are extensively working on the health of these animals. The outbreak of several viral diseases is a huge challenge which are faced by the aquaculture farmers. In one stretch, massive mortality is occurring. So while we can sequence the genome, of course, many of the fish sequences, including this oil sardine by CMFRA, SIBA, NBFGR, all of these institutions are sequencing several genomes of fishes, and these sequences are getting now, and mitochondrial genomes are also sequenced. But when we look at aquaculture, the disease is happening at the interface of the pathogen, the animal, and the environment. So, a kind of population medicine approach is based on our experience. We are proposing a kind of population medicine approach in which, when we look at the animal, the genome sequence of the animal is, of course, very important in marker-assisted selection, in understanding the disease-resistant, selection of disease-resistant varieties, and also to understand their adaptability, we have to study the transcriptomics. And we know that there is a stable microbiome in humans and animals, and it was doubtful whether these aquatic organisms have a stable microbiome. And we are going towards the idea, based on many of the metagenomic and microbiome studies, that they have a stable microbiome and a core microbiome, and dysbiosis do occur. And when we are feeding them with a certain feed and immunostimulants, there is a shift towards a favorable microbiome where there is less pathogen. So the presence of pathogen doesn't mean that there is disease, but the interface of all these are happening. So development of genomic resources is one next important step we have to do towards this aquaculture species, especially many different species of Kerala. Why it is important? Just like we know in human medicine also, we know because of the epigenetic factors, which is very important to have region-specific genomes. For this also, based on the environmental quality, the disease, the stress conditions, based on that, the health of the animal is affected. So apart from genome, so transcriptome studies are important. Microbiome studies are also important. Gut microbial health, and we have extensively worked on all the feeding trials which we are doing with fish. It can be shrimp, fish. All these trials we are doing now continuously. These microbiome studies also, and we are understanding that there is a stable microbiome indicating health. And when we have these genomic resources only, we can go for, the highly advanced tools like genome editing, to have this. So one work which is being done at our institute is knocking down this MSTN gene which is preventing the muscle growth. using genome editing so that there is more muscle growth, and this is being standardized in zebrafish and in other fishes also it is being tried. So for any of these advanced tools to use, we need to have the genomic database. And another aspect is that we have several, which was not covered in the previous section, is that when we are thinking about fisheries, we are thinking about only fishes, and this is a session on animals, but Government of India has recently given approval, and so much funding for considering seaweed as one component under fisheries. So developing seaweed genomic resources is also very important. And as it was already discussed, the methodological challenges are very critical in this aquatic field, starting from the DNA isolation to have good quality DNA to do the next generation sequencing. That is one challenge which we have faced. In fact, we are hosting a next generation sequencing facility at our university, which is funded by, our center is hosting that, and it is funded by Kerala government under KIFBI. It is an 8 crore worth facility which can be used, as one of the, apart from other commercial ventures, this can be used. So developing these integrated resources is very important when we think about fisheries. And now, of course, SIBA has generated the genome sequence of Etroplus. We have also done, sequencing of many of the aquaculture important microalgae, probiotic strains, and even this Etroplus. But since we have this Illumina Next 6 strain instrument, we could not have the complete genomic sequence.
Yeah. My next question is to Dr. Ramprasad. World over, a lot of sequencing is happening, gut microbiome, fecal microbiome, whether it is shotgun or metagenomics. And the other thing is that, all this microbial dysbiosis has been connected with many diseases. Like, for example, people are talking about gut-brain axis and also some central nervous system diseases, including schizophrenia. Now, as scientists, we will have to really evaluate. Sequencing is one part, great. But anything really proven beyond doubt, that's not the case with human genomics is concerned. You know that if SNP is identified, there is very clear-cut evidences are there. If mutation is identified, variants are there, it is. So variants are identified, that there is a lot of relevance has been implicated and it is there. But what about gut microbiome? What is your thought? Because it's really growing.
Yeah, I was little subtle in the earlier question when I mentioned that the near perfect correlations are not seen. Okay, I'll give you a small example. There is something called FMT. Okay, fecal transplants we do. In some of the severe dysbiotic diseases, you take inflammatory bowel disease, especially very severe young onset diseases. Now, one of the way of management is you basically at a young age you do a fecal microbial transplant. So you have fecal banks available. If I'm right, even Kerala, a couple of private fecal banks are there, maintained by some of the renowned gastroenterologists. So you have a collection of fecal samples which are characterized. Basically, you do a metagenomic sequencing, and you know in that what are the different species and what is the diversity that is present in that, and you store them and freeze them and store them. And whenever there is a case of acute, early onset, severe dysbiotic diseases which are there are few procedures are done where you do a fecal microbial transplant. Okay. Now there are decent papers published which mention that you do an FMT, there is an improvement in reducing the dysbiosis, and also in the overall gut health in such diseases. But not many randomized controlled trials which have clearly proven. It's more of an experimental currently happening. We have done basically we supported some of the hepatologists and gastroenterologists to characterize the fecal samples. We can clearly see when there is a dysbiosis, you see 90% of the whole microbiome from the fecal material of the affected individual has no diversity. So 90% will be only either dominated by a single organism, which is not a common one. The post you do a fecal microbial transplant, within about a week, for about 2 weeks, you see a good diversity that comes in, probably because of the transplant that happens. But most of the times we have seen after 3 weeks or 4 weeks, by the time you go for the second sampling, the diversity goes down significantly again and you see the symptoms coming back. Okay. Now overall there is a correlation, but precisely you cannot exactly tell with a precision this is going to happen. It is because of this particular species of bacteria. It is because of so much of abundance of this bacteria, you can intervene in this way. So this is applicable for majority of the microbiome applications that we are doing here. Now it can be because of the technology is still not that great to identify, especially the analytical methodologies. While sequencing with long read, you can characterize predominantly all of them, viruses and everything. But this is, I'm talking about stool samples where normal flora is there, or skin or any other non-sterile samples. But in sterile samples, it's fairly easier nowadays. If you do proper methodology, pre-analyticals are good, you can identify with a great precision the pathogenic bacteria present. It could be a CSF, it could be a blood. Most of the times you do a proper way, I would say compared to conventional methods, there is a significant increase in the diagnostic yield that you identify the pathogenic bacteria or a virus that is there. So microbiome is still, I would say, a bit flaky. A lot of methodology improvements, analysis especially. And today, how do you collect a sample? There's still sample collection methods are not great. How do you collect a stool sample? It's very difficult without contamination, outside bacteria getting in. So there are many things which needs to evolve.
You know, Yakult.
They're good source to identify many biomarkers, but again, the sample prep methods are very challenging. Contributing, yes.
You know, Yakult is sold something like 25 billion worldwide. And it has been mentioned that, you know, it contains certain organisms, mainly lactobacillus. And I have checked, I don't think there is no clear-cut correlation established consumption of Yakult to any kind of conditions. I have one more very, very quick question before going to them. The same way, vaginal microflora is coming up, and so is the skin microbiome. Especially that, you know, do you think that that will have a future?
There are some nice studies published, and even people do female reproductive microbiomes, especially we call endometrial microbiomes. You take an endometrial swab. It's highly correlated with fertility. There are several IVF centers as well as OBGYN centers which use endometrial microbiome to assess and tell. The higher the lactobacillus species, the higher the chances of conception. And it's fairly proven. It's not dicey stuff. It's still a good number of papers are there. Empirically today, many of them treat with antibiotics also or give lactobacillus supplements. Which is a science that is shown that there is a strong association between lactobacillus supplements with conception, especially in infertile couples who are there. So when you do endometrial microbiome, your lactobacillus species, when it's very high, above 95%, you consider it as a normal one. When it's low, it is considered as dysbiosis. and you treat them with an antibiotic or give lactobacillus supplements.
And also 2 weeks before the delivery, that women's vagina is full of lactobacillus. Is that right?
Yeah, the microbiome changes completely.
Okay. Now, one question, because a lot of urbanization is happening. And because of this urbanization, whether there is a significant diversity changes in the various type of microorganism, and is anything connected to, for example, the prevalence of kind of disease to get?
So this is probably I wanted to connect it with the previous session. We talk about diversity in terms of urbanization. The alpha diversity has come down significantly in urbanization. So when you take a human, they take a food, the food goes to the crop goes to soil and water. So it's always linked. So any changes that happens in that entire system is going to significantly affect the humans, gut microbiome as well. This actually, that's what the finding is. As of now, the alpha diversity has significantly come down, which favors infection to be more in the system, and that also causes inflammation and autoimmune disorders, because a normal child in a village will get exposed to maximum number of microbes in their early days of time. And here, with the growing awareness of microbes and hand washing, those things are all coming down significantly, and that you can also see how COVID response was there in cities and villages. By going by the trend now, even the type of food that we have and the type of vegetables that we have, this will change with the type of a soil we have. So the microbe, probably a vitamin or any growth factors or that is missing in the food that we are taking, which is going to significantly affect the gut microbiome, which in turn favors the growth of pathogenic bacteria. So it's not only that, even autoimmune disorders are also high because of this. People are connecting with this. So obviously, yes, there will be a change. By looking at a far ahead, now you get KFC in every place, and so the food habits also are changing. So we don't know how this will progress in due course.
Thank you, and we are actually running out of time, but then one question from our delegates. Anybody want to ask one question to any of the panelists, please let me know. If no more, then thank you all the panelists. Thank you very much. You have one more session which is going to come. Thank you very much. Thank you. Thank you, everybody.
Thanks to the wonderful panel here, Dr. Ramchand, Dr. Valsamma, Dr. Ramprasad, and Dr. Ananda Subramaniam. Tea is being served, so we are not taking a break, but in case if you want to have tea, tea is being served on the side. Our next panel will be on human genomics. Yeah, so the themes would be from DNA to diagnosis, bringing genomics into everyday medicine. And we have a wonderful panel here. So Dr. Shankar. So Dr. Shankar is a highly experienced medical geneticist and professor in the Department of Pediatrics at SAT Hospital, Government Medical College, Trivandrum, where he leads clinical and academic initiatives in medical genetics, growth disorders, skeletal dysplasia and hematology, with extensive teaching experience over 26 years. Leading human geneticist and CEO of Genome Labs, with over 2 decades of contributions to genomics research and innovation. With more than 59 peer-reviewed publications in prestigious journals, he has played key roles in advancing polygenic risk scores for South Asians, the Genome Asia 100K project, cancer immunogenic platforms, and other large-scale genomic initiatives. Then Dr. Shri Kumar, I also want to invite Dr. Shri Kumar. He's a clinician by training and is currently the founder, chairman, and director of Genome, a Kochi-based company advancing personalized health solutions through nutrigenomics and genomic science. He is also a part of Saukya Foundation and BioWell Hospital. Can you play the video, please? We have a video. So, Dr. Vinod Skaria was supposed to join us, but he couldn't be here because of some other commitments. So, there's a small video by Dr. Vinod which should be played, and after that, we'll start with the panel.
A very good morning to you, and it's a pleasure to be here, and unfortunately, I could not be there in person. Over the next few minutes, I think I will discuss about translating genomic data to clinical and public health insights with some cues on how the Kerala Genome Data Center could build upon some of these concepts. Now, when we talk about genomics per se, to me there has been no other technology in the recent human history that is reduced by 1,000,000-fold in cost, with consequent significant, almost exponential increase in the throughput. And this would mean that sequencing a human genome, which used to be an exotic scientific expedition 2 decades or so ago, could now become a routine clinical test, and in the future probably be available to every individual in the country and in the world. And there's also been no other technology that has been rapidly incorporating evidence to advance human health. Now, there's no other example that we can go back than the recent pandemic, how genomics can actually change the way of traditional ways, or change the way how traditional microbial discovery and characterization could happen. In fact, if you have been following the discovery of the Wuhan virus genome, which was sequenced from Wuhan during the outbreak, it was very evident that you could sidestep all conventional mechanisms or methods of microbiology to be able to sequence a sample directly from the infected human to be able to sequence the virus. And it's not just about sequencing the virus to diagnose the condition, but also to be able to characterize it genomically, to place it in the context of the evolutionary history, and be able to say that this is indeed a new emerging virus or a new emerging pathogen. Now, from one genome, this could be implemented at scale, and this was possible through the COVID-19 Mega Labs, which was an initiative of the Council of Scientific and Industrial Research, which I was fortunately part of, to be able to scale from one sequence to a few thousand sequences, to be able to use genome sequencing and genetic epidemiology as one of the mainstays for public health decision making. And it was Kerala which was the first state to sort of implement this as part of the public health decision making program, thanks to Dr. Rajan Opragade, who took initiative to essentially set up a statewide genomic surveillance program involving multiple districts, surveillance centers, universities, and research laboratories. And over the pandemic, this could provide very useful insights into understanding emerging variants, in our ability to predict new outbreaks, in our ability to characterize new outbreaks, and more importantly, to provide key evidence for public health decision making. And in fact, apart from public health decision making, this has also contributed quite an enormous insights into science and advancement of understanding in our ability to look at immune escape variants, emerging pathogens, and emerging lineages of the virus. The other major application is to understand prevalence of genetic diseases. While we call many of these genetic diseases as quote-unquote rare genetic diseases, what we today understand is that as we sequence and understand more, largely from population scale genomes, Many of these genetic conditions are indeed not rare, but rather undiagnosed and underdiagnosed in the population. So genomics at a population scale can enable the right evidence to enable clinicians to diagnose patients with genetic conditions very early, very efficiently, and also be able to treat them appropriately. I'll probably just give you one example of how this was possible with Indigen, India's first population scale pilot genome sequencing initiative, which sequenced around 1,000 odd individuals from cosmopolitan populations across the country to draw up prevalence estimates for some of the underdiagnosed genetic conditions. And one such genetic condition is primary immunodeficiency disorders, quite very prevalent in Kerala. And this was work with Dr. Geeta Govindraj, who is also my teacher at Calicut Medical College, where she worked towards using a multi-pronged approach to set up a screening program, a diagnostic initiative, and of course a research program, and also build an ecosystem where children with primary immunodeficiency disorders can be diagnosed early, treated early, and more importantly, can have a lease of life. And in fact, with support from the Kerala state government, along with support from the funding agencies and not-for-profit entities, including FPID, we could create a center in Calicut Medical College to be able to diagnose patients with primary immunodeficiency disorders quite very accurately and quite early. In fact, a significant number of individuals could avail treatment through intravenous immunoglobulins, which is supported by the Kerala state. Some of them could access curative therapy, which is a bone marrow transplant, along with support from the Kerala state government, and many more could prevent the disease by early genetic counseling and rather evidence-based genetic counseling. And given the high prevalence of the disease currently, with support from the state, we are also running a pilot newborn screening program for primary immunodeficiency disorder, the first state in the country to be able to implement this, at least in one center in a pilot process. And in fact, there are innumerable children who could get a new lease of life through genome sequencing at the right time, at the right place. And there is one such child who was also covered quite extensively in the news because his child could survive with the right diagnosis and a whole genome sequencing and also avail hematopoietic stem cell transplant, what we call a bone marrow transplant, at the right time. And genomics today can enable diagnosis quite very fast. And in fact, this is data from Seattle children which appeared last year. And in 2022, Seattle Children's instituted a process to get away with all standard of care genetic testing and replace it with whole genome sequencing. And the outcomes have been quite remarkable that they could reduce the time to diagnosis from 280 days to less than 15 days. And I think there is nothing more remarkable than this in our ability to reduce the diagnostic OTC of patients with genetic conditions, and many of them would lose the window of opportunity with a delayed diagnosis, with multiple tests, piecemeal approaches, and of course, unnecessary tests. If you can sort of merge this into one single concept of using genome sequencing, we can reduce significantly the diagnostic OTC for such children. And the other application is the ultra-rapid sequencing in newborn ICUs, where sick children in the newborn ICUs who would require a very fast diagnosis could avail this by using ultra-rapid whole genome sequencing. And this is currently available in India. In fact, you can get your diagnosis much faster, saving not just the life of the child, but also quite significantly saving money for the parents, the society, as well as for the governments. And as it becomes cheaper, I think whole genome sequencing is going to be impacting all of you, not just patients, but apparently normal individuals like you and me who may not have an apparent genetic condition. And in fact, this early data from the Indigen program sort of suggests that 1 in 20 individuals of apparently normal individuals in the population could have a genetic condition which is clinically actionable. And roughly 2% of them would have a cardiac disorder, cardiac channelopathies, cardiomyopathy, or familial hypercholesterolemia, which have current guidelines on treatment as well as prevention, or another 2% could have a higher risk of developing cancer, including breast and ovarian as well as colorectal cancer. And given that Kerala has a very high incidence of cancer currently in the country, I think this is probably the way to go to not just reduce the mortality due to cancer, but also enable early diagnosis and institution of care with much better outcomes compared to late diagnosis. That is the norm today. Now, having said that, this will all culminate in the ability to convert healthcare from treating the illness to promoting wellness. Because all of modern medicine starts with a patient, and a patient is by definition who has symptoms and signs. If our ability to diagnose an individual before the onset of symptoms and signs would necessarily mean that we can prevent or delay the onset of the disease in a considerable number of individuals. And this considerable number of individuals with new therapies being available is very, very large. And I think the future is our ability to be able to move from illness to wellness and move our ability to diagnose a condition from symptoms and signs to pre-symptomatic, if not an asymptomatic individual. And this is today possible by what we call as newborn screening. And in fact, newborn screening could also be broad-based to encompass a genomic newborn screening. And in fact, some countries are already implementing a pilot genomic newborn screening with our ability to diagnose preventable, highly penetrant, with high evidence of actionability for treatable conditions which could be treated with an accessible, affordable drug or therapy. And I think as we move into the next 1 decade, this is something that we would increasingly see as a mainstay, given the large evidence that is emerging in the recent years on the diagnostic yield of genomic newborn screening. Now, one thing that we need to keep in mind is our genome is constant from the day we are born to the day we are dead. But the ability to answer different kinds of questions in the context of your lifespan is quite remarkable. At different points in your life, you could ask very different kinds of questions and also be updated on the current evidence without having to resequence yourself. In other words, genome sequencing is for a lifetime. I think with that, we'll stop, and not to exemplify that Kerala Genome Data Center is at the right place at the right time, and of course run by the right population of people and with an ability to impact the state of Kerala, not just the state of Kerala, but also to accelerate discoveries, and not just in India, but also for the rest of the world. Thank you very much. I would be more than happy to take questions. You could reach out to me on social media or on email.
Yeah, thanks for that talk, Dr. Vinod. So, for me, this panel is quite important because human genomics has reached quite a lot of innovation in the recent times. And we have two clinicians here and one person from the diagnostic space, MedGenome. So, my first question will be to Dr. Shankar. He's a pediatrician by training and has been pioneering medical genetics in the state of Kerala. Sir, at what point should a pediatrician suspect a genetic etiology? That will be my first question. And when is the right time to send that sample for sequencing? And in the current context, in the set of patients that you're treating, because you are from a government institution, are we currently under testing or over testing when you look at both the private and the government setup? So what's your opinion on that?
Thank you. Thank you, Dr. Raju and KDC for this opportunity and the fabulous opportunity to have a genomics in Kerala as a Kerala Genome Project. Because I have completed my DM in 2007 and I came back on last 16 years I am practicing genetics without even NGS. NGS came in 2013-14 and so in the first era practicing genetics was very, very easy. Because we are having few genetic disorders where diagnosis can be confirmed. So the first question what Raju asked, which is Dr. Raju asked, is very pertinent. When a pediatrician should refer to a geneticist? because you suspect a Down syndrome, you suspect a Turner syndrome, multiple malformations are there, thalassemia, hemophilia, SMA. There are specific monogenic disorders and chromosomal disorders where genetic testing was possible in early part of my career, or you can say before 2013 or 15. After that, when this NGS came, exome came, people are doing genetic testing rampantly. So last question is already answered. It is actually overdoing to certain extent. But really in some patient where it is needed, it is not doing, there it is a underdoing also. Because that clinician is not aware that this is a genetic disorder and genetic testing should be done. But at the same time, in the other part of the story, because of the various availability and various pressures, some people are doing overdoing also. Because people want to know that if epilepsy is there, okay, we will do it. Neurodegeneration, we will do it. So as a clinician point of view, first we should analyze what is the analytical validity, then clinical validity, as well as clinical utility of the test. All genetic testing cannot be, as you know, most of the genetic disorder is not curable. Even if they have done the test with 15,000, 20,000, 30,000, you cannot do any cure, only you can do genetic counseling. So selection of the patient is very, very important, and the clinician should be aware of the when you have to do it. Where you will do it, there is a lot of areas, because if you take system-wise medicine, if you take hematology, lot of anemias, there is resistant iron deficiency anemia, hemolytic anemia, Fanconi anemia, plastic anemia, bleeding disorders. Most of these conditions you require genetics. If you come to cardiac evaluation, some of the complex congenital heart disease, and congenital heart disease is a part of a syndrome, cardiomyopathy, arrhythmias, where the channelopathies is the major important thing. Neurology, there is no limit. Endocrinology, there is no limit. So in any genetic conditions you do, oncology, there is no limit. So definitely number of genetic conditions are increasing. Clinicians should be aware, but I am very skeptical, very, what you can say, very unhappy that in this big crowd, only few clinicians who are working in genetics are only there. Because clinicians will not have that interest in genetics. What they feel, that's I told, in previous era, before NGS, if somebody told the clinician, they don't want to take that interest. Now NGS is available, they felt that only one solution is NGS. Just like hemoglobin, they will write NGS and they will send. After getting the result, if some confusing things are there, they will refer to a geneticist. So actually the situation is little bit complex. That means underreporting in one side and overdoing in other side. And at the same time, I think a good practice in genetic testing should be taught to the clinician. I think this human genome project, because here, what is the major constraint before that? It's the cost. This cost was very, very high for exome. In 2017, it was 40,000, something like that. Then it has reduced. Now it is reduced, no doubt about that. But still, it is a little bit high compared to other tests. So, one group of patients, really it is required, they are not able to do it, even if it is required. But other group of patients where, as a fashion they are doing, that is also is there. I will just identify some problem is there. Okay, I am talking about the genetic disorders and rare diseases. If your clinician's perspective, genetic disorders are two groups. One is rare disorders, monogenic disorders, chromosomal disorders, hereditary cancer syndrome, where genetics do a lot but non-curable. Other side of the story is a polygenic disorders like diabetes, hypertension, coronary artery disease, schizophrenia, asthma, where there is a susceptibility gene is there, but it is not the answer. Similarly, the wellness genomics also. So they are doing a lot of wellness, that I will come to that later. So these type of areas, you can do it, but proper counseling. So all testing should be pre-test counseling and post-test counseling is required. Whether that is aware by the clinician or whether they are having a time, lot of questions we have to answer. I think this is the introduction. Okay.
Yeah, thanks.
Okay.
Thank you, sir. Now my question will be to Dr. Shri Kumar. So you are one of the pioneers in nutrigenomics in India. So you started with some of these tests and the analytics way back in 2000s. So my first question would be, what areas in nutrigenomics are strongly evidence-based? Because for clinicians, a lot of companies will come with tests, but how much of them are actually evidence-based? And where are we still in the early days of, where this can be ahead of science, where you can actually give a good diagnosis or the interaction where patient can benefit?
So thank you, Dr. Raju, and thank KDC for giving this opportunity. First of all, this question, especially, and Dr. Shankar has answered one question. Based on genomics, you cannot do any treatment. But we know one thing, genomics drive disease. So what are the thing, because especially when I started on genomics, especially on nutritional genomics, I started in the way much beyond, because I started testing this. It was not available in India, it was available in Australia and US, so I was doing it. So at that time I could understand that, okay, there is no medicines or no counseling or no other preparations available to treat. So we have adopted a module. So we try to link the genomic expression with the metabolic disorders or metabolic findings. So we have found a lot of things that is coming in. So it is actually balancing between the genome and the metabolism. So this is where it comes. So we have, I think now we have thousands and thousands of cases now we are documenting and we have to publish it. So only thing is that because now you can see that, okay, this genome is there, that may not be applicable to one diabetic patient or one cardiac patient or one cancer patient. But if the metabolism is there, you can see this genome is responsible for making this metabolism to happen. So this is one of the end results. But again, when we also switched over to microbiome in that extent, we understood another factor, and that is actually way ahead of our genome. So microbiome is actually different. So now we know that, okay, you have to understand the balance between genome, metabolism and microbiome. But again, that was not giving the answer, as Dr. Shankar was telling. Then everything is, I think some of the speakers had told before, it is also the environment. So now we have come to a conclusion of how to bind or how to bridge the gap between our new genome sequencing, then comes the microbiome, and we call the environmental exposure, we call it as exposome. Exposome has also become one of the lifetime environmental exposure which is causing some of the effect in your body, and all this can be balanced together. So we have lot of things, and I think Dr. Vinod was talking about illness to wellness. We were doing that for last more than 15 years. I have more than 15,000 patients now living with illness to wellness. So we have done a lot of conclave based on that illness to wellness conclave in Cochin. So we have understood that, okay, thousands and thousands of patients can be benefited. But how we know? We look at the balance between this, this genome and the microbiome and the exposome. So finally we can say there's a combination of all omics, nutrigenomics, pharmacogenomics, exposomics, and finally it should be metabolomics. As Dr. Shankar was saying, no clinicians are here, but clinicians should be taught, especially they should know about all these things. They can link. Only clinicians, if they come forward, we can link this to multiple, especially that is the forum in which we are sitting. Maybe this can influence many clinicians to come forward and do this testing. And I think this is going to be the most scientific, advanced and evidence-based healthcare practice maybe in the today and also for the future.
Thanks, Dr. Srikumar. My question will be to Dr. Ram. So my question will be on the cost side, because typically for most of the genetic tests, it's not accessible to a lot of people because of the fact the cost is very high. When we sequence the first genome, the cost used to be in the $1 million range. Now it's reaching somewhere around $1,000. And as a company who has been in this space, what can help companies like MedGenome to reach that to a less than 10,000 mark in India? And how can it sustainably do that?
So you mean to say a whole genome, clinical grade whole genome at 10,000 rupees?
Yeah. Or maybe similar tests where there's a lot of actionable insights which can come up.
Oh, exome, a good exome is pretty decent nowadays. Quite actionable. It's less than 10,000 rupees. It works. As Dr. Shankar mentioned, 10,000 itself is very high for many of the patients. Okay, they can't even afford 5,000. That's why I was telling these initiatives like this or any government support programs are extremely important for those patients. So coming back to the cost, sequencing costs, Currently, there is a war going on, sequencing war. So if you look at, we're talking about $100 genome. Last AGBT, just a week back, 3 companies have launched a $100 genome. Somebody has shown a roadmap for a $20 genome. We expect it's beating the Moore's law. Costs are going down. But one important thing which everyone is forgetting is, for us, today sequencing is not the highest cost. For us, data storage and data analysis and how we handle the data is costlier than sequencing. Just last year, it crossed. Okay, so today you want to either analyze the data, it's a compute, whether it is a storage or whether it is sharing or whether it is how it is handled between collaborators and partners. That's the costliest part of it in the whole of the ecosystem that we do on genomics. So sequencing costs will come down. They will become, now sequencing costs are so low, some of the conventional biomarkers and conventional testing is expensive than doing sequencing. Okay, so sequencing is a commodity. To be very frank, in large initiatives, we shouldn't too much focus on sequencing. What we should focus is down the lane what comes. How are the costs handled? How government is going to support all the stakeholders with such heavy investment costs which are there. These are all, data infrastructure is an upfront cost, is a significantly high cost. So sequencing costs will keep going down, become mature. Today we're talking 5-base chemistry. We do methylation along with regular sequencing. Today we're talking long read sequencing. So more and more varieties, more and more stuff will come, but costs will keep going down.
My next question will be to Dr. Shankar. This would be on the variant interpretation. So we also see in clinical practice interpreting variants of uncertain significance, right? You may get. So how do we build local expertise in interpreting these, some of these stuff coming up?
Okay, that's very difficult question, because the nightmare of exome when we are sending, or a genome we are sending, is the VUS. If lab give a VUS, it will be a big major headache for the clinician, because I have to tell the patient that one is, it is not that significant, but it may be significant. It may be a cause for the disease. We cannot do prenatal diagnosis based on that. We cannot give a molecular treatment based on that. Then they, okay sir, I am normal. No, that cannot be made sure. And researchers will say that we can reanalyze it, we can do that, we can wait for some more database. For a clinician, VUS is a big headache. Big headache. So I am little bit worried about the exome, sending an exome or genome. Genome, as you know, most of scientists know, genome will give a more VUS. Exome, if you do clinical exome or VUS, will be less. That is why. Genome means lot of VUS you will get. So many variants you will get. So I am little bit worried. This worry is not for a regular clinician. That's why I told. For a neurologist and gastroenterologist, they are not bothered about the VUS or other things. So they will send the exome. When VUS they will get, they will refer to us. Then our job is to settle the VUS. So VUS is a very much big headache. In the clinical practice, my major hard handle for VUS is clinical phenotype. A researcher at the lab, he will not get that. I am seeing the patient. So I know that you have given a VUS. I am 100% sure the phenotype is not fitting you. As a medical geneticist, I know that it is not definitely. Today morning, just before coming here, a patient came, it's a 36-year-old female, pregnant, referred from Shri. Their clinical diagnosis is hereditary spastic paraplegia. I also felt hereditary spastic paraplegia. And the lab has given a report that it is some mitochondrial things, heterozygous VUS. Something like that. So they have given, because they have got some phenotypic fitting is there, they have given. But I am 100% sure it is not a mitochondrial disorder. It cannot be, because it's totally different phenotype. So that phenotype is one thing which will help me practically do the counseling. I am not telling I will immediately change his VUS to likely pathogenic. That I cannot do. That multiple testing and other things are required. But for a practical way, one is phenotypic thing. Second thing is we will do parental segregation and we will try to settle it out. Third most important thing is database. If database is good, your VUS will come down. Naturally, because we are using a external database, it's a problem. Asian database is Medgenome or Newberg is using, their results will be little better than a third lab, they are not having that personal experience because they have done more than, I don't know number, so much because I am starting from Genome, I am sending some samples to Genome. No, one of the big company. So they are having large number of samples. Their results will be a little bit because the database is more stronger. So when database is stronger, result will be better definitely. But that is the only that is the only good answer. So many other things like parental segregation, other things, etc. is there. So VUS is a nightmare, which somehow this database will help you to answer.
Thank you. Dr. Shri Kumar, next question will be on metabolic disease burden. So Kerala has a very high burden of metabolic diseases like diabetes, hypertension, dyslipidemia. So can gene-diet interaction data meaningfully alter diabetes prevention programs or maybe hypertension prevention programs, etc.?
So one of the things that we have to look here, because if you look into the microbes around and if the food is that is available, it also, if it is giving actually growing in the atmosphere, the genetic expression change with the environmental change. And we are fast changing. So one of the things that can change is only the gene expression. So gene expression of the food that we are consuming that is coming from outside, that will change. But only thing is that we have to see that, okay, that is balancing with your genome. So one of the greatest thing that is happening today, so when you talk about the metabolic disorders like diabetes, heart disease, cancer and all these things, we know that the basic pathology behind all these things are inflammation. So the inflammatory disease is going high. So the inflammation is caused by various, maybe it is, when we know that it is caused by various immunological or metabolic anomalies that we can see in the blood biochemistry and other things and inflammatory markers. But if you go into the genes, so if you look into the pathway between the genes to genomes to inflammation to the disease production, you can see there is a huge track that is coming in, and only thing is that our aim is to block it. So one of the thing is that is absent now is, as Dr. Shankar was telling about, is the lack of the database. The database should be there in one, whether you do it in this lab or that lab, there should be a database which should be there. And based on that, we should also know about the microbiome and other things. So genomes, gene, diet, experiment. So we call that type of diet, especially today we are giving a diet. If you ask a dietitian, they are giving a diet for diabetes, heart disease or cancer, whatever it is, but it should be for your personal or personalized diet. So that means if it has to come, we call it, it's a diet for your genetic expression so that it can manipulate your metabolic or inflammatory change. So this is possible. We have seen we have a very strong connection between various dietitians, nutritionists and other researchers. So we are seeing that, okay, we call this epigenetic diet. That means a diet for your epigenetic expression, and that can, then we can look into the various metabolic factors that is changing over the time. So for a long duration, maybe for when a patient comes for treatment, you can use all the drugs that is involved, but at the same time, if you understand what is happening behind, that is with the genetics, genome, gene expression and metabolism, and if we can manage it with the diet, so that is going to give a long-standing health, and that is why we call today it is a precision health, which should actually come for, and then when you manage it, we call it as precision medicine. So for precision health, we need precision nutrition. And if this has to happen, we should actually scientifically collaborate this with, and this is why the government forum should be there. And I think especially when the gene tests and the tests are very costly, I think just like in US, we should try to incorporate insurance into the gene test. That is actually absolutely essential for it, so that financial this thing can go. Only then it can become a widespread gene testing that we can do, and state, especially local. Because you can see that I have seen, we have done a lot of studies, especially vitamin C manufactured in USA, vitamin C manufactured in India and in some other country. You compare all these things, you can see the polyphenol and the micro analysis will say that, okay, maybe it is containing 500 milligram, but the effect is different. Why that is different? Because it has to go with the local. So if you are manufacturing a vitamin C which is sourced from the US, it may not work here. So you have to get a local thing to manufacture things. So that is, we used to predict one thing, eat natural and eat local. So it should not be, you should not eat an apple coming from Kashmir or Canada. You have to eat an apple from Munnar or Kodaikanal or Ooty. So that should be, you have to have a local instinct that can only support your genes because your genes are living in this world. So a diet and genes have got a lot of things to say, and it has to say basically on the metabolic happening inside all of us, and that can actually lead to health actually improvement. So what we do is actually doing a improvement of the health or empowering health and empowering the immune system. So you have to have there. So that is why prevention is not preventing disease. Prevention is empowering health or maybe strengthen health more and more. So that should be the way to go forward. So I think that answers your question.
Dr. Ram, the next question will be, how can private genomic organizations like yours collaborate with state initiatives, national or state initiatives, without compromising on data privacy and public trust? Because genomic data is valuable, right? And anything which goes can have a lot of implications. So what do you think can be a good way to build that trust and collaborate?
See, one important thing for scaling up omics in the country that's missing across the country is we don't have ethical and legal framework for data. Okay, I know central government has come up with Digital Data Protection Act. It's still 2027 is the time when they will execute it. I'm not sure whether we are ready with the infrastructure or not. But currently there is no clear, legitimate and very simple digital data framework, both in legal and ethical aspects. Give you a small example, Europe has something called GDPR, General Data Protection Act. So that, at least if not in that detailed level, some kind of a framework should be there for not only just private and public, for any stakeholder in the ecosystem to work in a proper way. That's the most important thing, because interoperability as well as data sharing as to what you disclose, what you don't disclose. So ultimately what happens when you don't have this framework is it's like free for all or ban for all. Either everyone does everything or you just say just stop everything. So that is something I would say one of the most important things that today we need to have for these collaborations.
Yeah, so any questions from the audience, because we are running a bit late on our session. So if any questions from the audience, maybe we can take one or two questions. Yeah.
Yeah, particularly to Dr. You are talking about a condition where you had a different clinical outcome, but laboratory diagnosis was this. As a researcher, we would be interested to know why that things happens, and there is something, a message is being passed which we are not able to pick it up.
That is the thing I told. As a clinical point of view, now that gene or that variant is not explaining the disease. So there are two possibilities. One is the actual cause of this disease is already there, but we have not identified. For example, in HSP, hereditary spastic paraplegia is having more than 30-40 genes are there, and in some of the genes there is exonic deletions are there, so NGS may miss it. You have to do MLPA or deep intronic mutation.
That is fine, but mitochondrial changes that has happened.
No, that's it. In mitochondrial, there is a variant is there. That variant is a VUS. That may not be significant. That's the thing. I told one is, so there are two of them. Second I have not told. First thing is, we are missing the actual variant gene. Second thing is, this variant, whether it's causing a different phenotype, that is also possible, as you told. But then there should be, evidence should be there. Without evidence, we cannot act. That's the thing I told. Researchers and clinicians' point is a little bit different. For a clinician to act, we require a concrete evidence. As you told, correct. After 5 or 10 years, sometimes somebody will tell that these variant has seen in this much of patients. All patients are present with the similar to hereditary spastic. Agreed, I will change my mind. Not my mind, actually that is changed. Evidence has been changed. So we can do that. But at present, there is no evidence. And without proper evidence, VUS, we cannot do actionable procedures. Two things, one is prenatal diagnosis cannot be done, treatment cannot be done. So in Western countries, as Ram Prasad has told, in these type of situation, VUS they will not report. They will not report the VUS in prenatal sample. They will not report VUS in the carrier analysis sample. So what has to be reported? If you identify, you have done for something and you have identified that the patient is a BRCA1 mutation, later they can develop the tumor or malignancy. In a 5-year-old child, they should report or not. If they report, I will counsel or not. Both there is no framework. That framework should be there.
Thank you. Any other question?
Yeah, any one of you can answer. It is connected to retinitis pigmentosa. What I understand is that, only one mutation, which is probably that, genetic treatment, I understand, Novartis has got. And as far as India is concerned, it is 1%. That means 99% will be other mutations. Now, but by identifying the other mutations, is there a possibility, some kind of treatment? I know that there is no drugs, no genetic treatment is possible, but some kind of supplements, drops, or to control the alterations are, it is possible.
Yeah, so retinitis pigmentosa or RP, it's a very highly heterogeneous disorder. There are more than about 140 genes, monogenic genes, which have been identified. In 1999, RP65 was one of the first gene to be identified in a family in Madurai. Very few people know about this. It's a consanguineous family. The gene was identified RP65, identified by a group in Chennai. And in 2015, first FDA approved treatment that came from Novartis. Okay, many think that Novartis did it, but actually the clue came from India. And they identified this drug. It's called, I forgot the drug name, but it's an RP65 gene therapy. It costs $800,000 per dose of treatment. You administer it very early in life, similar like SMA. There is some vision that comes back in several of them. But that patient has to be RP65 mutant. Then only you can give the drug. The RP65, the percentage of mutants causing RP is less than 5%. So 95% of mutations come from rest of the 140 genes. Okay. Now similar strategies are deployed, for example, RPGR. There are about a host of half a dozen genes. They have crossed phase 1 trials. They have been getting into phase 2 trials. But please note, these treatments are super expensive. Rare diseases, market is small. Pharma companies don't supply to countries like India where there is no insurance coverage. This is the biggest barrier for treatment currently. I'm sure Dr. Shankar knows more on SMA treatment, how challenging it is today. But there are more cheaper options, generic options coming up. We only hope in the next 5-6 years, these gene-based therapies also, generic formulations come in India with the cheaper versions, and it will be available for many.
And for RP, we will do genetic counseling, like anticipatory, what are the problems. Then in the other family members, prenatal diagnosis can be offered. And for RP65, it's a typical phenotype that's called Leber's congenital amaurosis. That is having a blindness from the early age onwards. Retinitis pigmentosa, totally different type of phenotype. Phenotype in different conditions are different.
The bottom line is that, if it is RP65, there is some treatment, there's genetic treatment. Other than that, nothing is possible.
No, that is the thing. Nothing is possible is a major mistake that I will tell. Because people felt that treatment is cure. No. Treatment is cure, control and care. Diabetes, what are you doing? You are curing diabetes. Most of the time you are controlling the diabetes. There is called a 3C concept. 3C concept means every management of a patient, when you are a patient, they come to you, there are 3C. One is cure. Few patients can be cured. Control. Most of the patients can be controlled with the symptoms. Third, 100% of the patient can be cared. That cared is very important for retinitis pigmentosa. What are the options available? What are the things are available? What is the chance of progressing the symptoms? What are the chances of developing to others, family members? Any prenatal diagnosis options are available? So we cannot say nothing is possible. We cannot cure the disease. That's all.
Yeah, which I understand. What I'm telling is that how to manage them. Other than RP65, what kind of supplements or I know drugs are not there.
That we have to counsel, we have to do whatever management possible options are there. Depending upon the conditions, it is different. For example, retinitis pigmentosa, I will told 100 heterozygous type are there. Different people will have a different type of problems. That an ophthalmologist will, depending upon the problems of the patient, will manage it. Some people will have the progression of the symptoms can be halted. So many things are there, but there is no cure except RP65 and few genes. Now phase 3 therapy studies are going on.
So there is one more thing, especially retinitis pigmentosa, because there is no cure and other things. Lot of people had come to me. So we have what we are, because we cannot do anything on that. But what we have done is because we have to, as Dr. Shankar said, we have to take care of them. So for taking care of them, we have done their metabolic profiles. So we manage their metabolic balancing. So we call it rebalance the metabolism of that person. So molecular metabolism or cellular metabolism. So when we balance it, they get better. Maybe it can be halted, or the further complications can be prevented. Other anomalies can be prevented. And we have, especially I think for the last 20 years, I have seen more than 100 RP cases. I think 2 or 3 patients at least can see something. So they can move around and they cannot read anything, but that is the only thing that has come. Maybe it is because the metabolism has been changed. So it is not because of the disease process. So taking care of the person is actually possible if you can really balance the metabolic profile of the person.
Last question to the panel would be on skilling side, because we have a lot of young students here. So as the number of tests increases, right, there will be need for trained geneticists, counselors, etc. So maybe both Shankar and Ram, what kind of courses are needed to train them, and what kind of skill set currently exists in the country, and how do we strengthen that?
The clinical point of view, as rightly pointed out, more number of geneticists are required, and I think definitely it is increasing. In 2007, when I finished, I was the only person in Kerala with the DM in medical genetics. Now we are having around 6 or 7 people are there, various parts of the state. In Trivandrum itself, 4 people are there. My students only, they have just completed the DM and joined. So there are so many genetic DM, that is the medical geneticists are increasing. Another major area is the genetic counselors. Genetic counselors are very, very important, because all specialists, especially the oncology and this type of specialty, one entire medical geneticist may not be required to see all patients. That type of, because these genetic counselors cannot diagnose the condition, but if somebody has diagnosed and identify a mutation and this is the gene, they can counsel. So genetic counselors are a major area where people can venture, and there are 4 or 5 institutes in India has started the genetic counseling courses. In Kerala also, I think genetic counseling courses has to be started. We are having few genetic counselors, but we have not started the genetic counseling course because of some administrative and other academic reasons. So genetic counseling is definitely has to be increased. Then already I told healthcare practitioners and other people's awareness should be increased. One is geneticist and genetic counselor from the clinical side. Lab side, I think Dr. Ram will.
So there is a lack of skill set here, trained skill set, competent skill set. We get on average every day about 150-200 applications, and still we need people. Okay, so there are 3 levels of skill sets we look at. Not only we, anybody who works on omics data broadly. One is pure software professionals who know coding, few languages, who have logic, who can think, who can code, design pipelines, write codes, proper software. I think there's quite a bit of talent pool available out. The second is who are at the interface of bit of a coding and quite a bit of biology. We call bioinformatics. They are not pure software coders, but they know how to write small codes, understand biology, put some scripts and run data automatically. We call soft pipelines. This is a skill set which is competence skill set lacking. Okay, because the biology is changing fast, complexity is changing fast, the automated analysis requirements are increasing significantly, speed needs to be faster. So we need people who can do this. Not only we, as a country, as a state, KGDC needs a lot of people like that. So today, there are good things about specialized training in colleges, universities, but one of the limitation is they are not industry ready. Okay, so two things that can be done. One, the students proactively, enthusiastically should jump into interns, learn things hard way, put their efforts and learn. Universities also can take some issues. So as a private company, for bridging the skill gap, we have one course just about 6 months back we started for primarily on the more on the interpretation side, whom we train on software to interpret the data. We do a batch of about 20. It's a 2 months course, a short course. They have to pay for it. It's not free. And at the end of 2 months, there will be a competency assessment, 2 competency assessments. Whoever passes it will be taken into the company. So I'm sure many other companies have such courses, private courses, but like it or not, the skill set gap is large for us. We keep on looking at, and now with the AI coming in, how to process GPUs, how to code, do machine learning stuff. It's a separate niche area, growing fast, but an initiative like KGDC has to bridge that skill gap, otherwise it's going to be difficult.
And one more thing I want to add to the KDIC, because it's a government initiative. I am also from government. I think we have to think positively to generate some post for this genetics and genetic professional. Because I am having a lab, I started in 2007, now I am having 15 lab technician, genetic counselor, scientist, etc. But not even a single government post. Not a single government post. As a person I am telling, I will tell that, okay, you are having faculty they will give. One assistant professor, faculty also they will not give. Because you know, medical college faculties, 1976, same number of faculty still is there. But most important thing is, this system is not there. There is only lab technician or senior technical officer. There is no cytogeneticist, no bioinformatics. If you give in the project, the office people ask, what is bioinformatics? They don't know what is bioinformatics. So problem is, everything is run by the project only. I am having DBT funded, ICMR funded, DHR funded. So many projects are there which I have done for so many years. So it's on my initiative I have taken, and 15 people came and I was somehow running the genetic lab. So my request is, since you are training these people, you have to tell that government post for this genetics, because genetics is not before 10 or 15 years. Now you require cytogeneticist, molecular geneticist, etc. In government side, post has to be created. PhD also has to be created in the government medical colleges. Because now medical college is different, researcher is different. What clinician is telling, researcher cannot understand. What researcher is telling, clinician cannot understand. This researcher and clinician gap is a very big gap. Only very few people who are a clinician-based researcher or researcher-based clinician, whatever thing we can connect. We can connect it, understand the language of a researcher, and my clinician and researcher can understand. Dr. Shri in virology, so many collaborations we are having with the RGCB, so many research scientists. So these two gaps we have to correct. Then only the medical colleges will be hub of the research, and more number of these type of people can be absorbed there.
So that is very, very significant. Especially you have to skill them in the technology side. Then the skilling has to come down to the clinician. Then the clinical knowledge has to go down to the people. So we have actually, this is the actually the interface. I think that is what the KDC and all these people have to. I think Raju has got a big task in front of him. So he has to make. Actually, we have done this some few years back in association with the National Human Rights Commission of India. So we have been taking this Genomics to the village. So we have taken 3 villages in Kerala and we have done this, a fantastic program. We called it and it is presented to the WHO and UN. So it was the Supreme Court judge who has been the convener. And what we did is actually awareness program. We covered around 250,000 people in 3 villages in Kerala. So that means, so the Human Rights Commission has got only up to one area they can fund. So that means only creating awareness and creating knowledge. And other than that, they cannot serve. So what we need is actually the course in the upskilling and also in the course in the lower level also, because now we are having starting a course especially based on the what in US they are doing. So we call it as culinary medicine. That is one of the things that is going to come. So that is mainly gene-based food, how you can give. And nutrigenomics, making into, so the term is something big, but you have to make it very popular so that the common man understands it. So there should be people who can talk to the, so we have in our hospitals, there are a lot of people who are doing it, converting that and telling the patients. So this type of people, so whatever technology coming tomorrow, whether it is AI or any other bioinformatics, any technology, it has to go from the research down to the people. So the clinicians are the people who are dealing with the people, so the clinicians have to understand it. So I will say, because especially Dr. Ram was here, so Shankar was here, so I am telling. So any MD or MS, and we were doing, it was not there. Everybody should do PhD. So only then the research should come. So without PhD, because that is what in Australia I am seeing, any MD fellow or any postgraduate or anybody who is doing, they definitely go for a PhD course. So when the PhD is being taken, the doctors will understand the language of the researcher. Otherwise, it cannot be converted. So we are creating, so Saukya Foundation, we are creating that platform from the researcher to the clinician to the public. I think in May we are doing a great function for that. So I think all of you are welcome. We'll intimate to you what are all the things. We are actually, so you need politicians, you need the policy makers, you need the clinicians, you need the experts, and you need the technologists and researchers, and everybody should be in one platform. This is for all of us. Thank you very much.
Yeah, thanks to this wonderful panel. So we go to the last section for today, which is on bioeconomy, skilling and policy. Thank you. Thanks. So now I want to invite the next 3 panelists, Dr. Naveen Anashwara, Dr. Sharika AR and Dr. Unnikrishnan. So Dr. Naveen is a dynamic public health leader and currently serves as the head of the Kerala CDC, which is Center for Disease Control. A medical graduate from Calicut Medical College. He served as a district program manager, National Health Mission Kozhikode, and he's an MPH from Johns Hopkins University and is currently heading Kerala CDC. Dr. Unnikrishnan is a principal scientist and head of operations at the CVJ Center for Synthetic Biology and Biomanufacturing at CUSAT, where he leads initiatives and collaborations bridging academia, industry, and innovation in synthetic biology. Earliest in his research career, he has contributed to interdisciplinary scientific work with affiliations including Duke NUS Medical School. Dr. Sharika is a microbiologist by training and a science administrator with over a decade of experience in shaping Kerala's science, technology and innovation ecosystem. She is a PhD in microbiology and scientist in charge of the Kerala Biotechnology Commission, KBC, the state's nodal agency for biotechnology promotion. She served as the project coordinator for establishing the Institute of Advanced Virology, Kerala's first dedicated virus research institute. So welcome to the panel. So my first question is Dr. Naveen. So you have been an administrator, a clinician, and currently working in public health. So what is something that you really care about? Like in public health, how do you incorporate genomics, genomic epidemiology, so that we can look at some of the core problems for Kerala, like the dengue, Nipah, etc. Right? So what can genomics add to that entire surveillance programs?
Thank you, Dr. Raju, for inviting me for such a wonderful platform. Now I am working as State Project Head of Kerala CDC, Center for Disease Control and Prevention. And as well as I am working for Nipah Research Center, Kerala One Health Center for Nipah Research and Resilience at Calicut Medical College also. So it's a relevant question that how we can integrate genomics to the public health. So here I just wish to introduce the term precision public health. Previous speakers already told about precision medicine. Now we are talking about precision public health. As policymakers, central government and state government, they are creating many programs and policies to prevent communicable diseases. And Dr. Raju especially mentioned about Nipah and dengue. So, how genetics is important in communicable diseases? So, I am starting with the Nipah. Nipah virus, there are two clades of Nipah virus, clade 1 and clade 2. And clade 1 is Bangladesh clade, and clade 2 is Malaysian clade. And unfortunately, we in Kerala, the clade is closely related to Bangladesh one. So it's more dangerous, and it's mainly presenting with more respiratory symptoms and high chance of infection, more than 70% mortality, and it's very difficult to control also. So there comes the importance of genetics. We have to do the sequencing of the virus, and we have to classify it, and it may change. Mutations are possible, and it may change from clade 1 to clade 2, and new clades, there are chances for new clades, and their character may change, and it can increase the pandemic potential of Nipah virus. That's why we have started a dedicated institution for Nipah research and resilience at Calicut. So we are trying to do a comprehensive program incorporating this genomic sequencing and all, along with the National One Health Institute at Nagpur and ICMR. And coming to dengue also, and hepatitis A also, the character of virus is changing, and especially in hepatitis A, you can see more young people are being affected with hepatitis A, and mortality rate is very high. So here we have to study what is happening in the genetic pattern of the virus. And so in the pathogen surveillance, in the public health point of view, genomics is very much important. And as part of Kerala Center for Disease Control, we are going to start a wastewater surveillance that through viral load assays in sewage water of especially hospitals, we are going to analyze the pattern, and we can easily predict the pandemics and outbreaks with this wastewater surveillance, and the genomics is the most important part of it, especially for COVID. These genomic sequence, whole genomic sequence in wastewater surveillance is very much important. And in the other aspects of public health, apart from infectious disease surveillance, newborn screening, already Dr. Vinod Correa mentioned, especially conditions like phenylketonuria, congenital hypothyroidism, we can easily do the comprehensive newborn screening. And coming to population genomic screening, we can screen high-risk populations for certain diseases and can take interventions. And when coming to pharmacogenomics, some drugs, they are effective to some individuals, and we can do these genomic studies, especially some examples like in colorectal carcinoma. If there is KRAS mutation, the treatment is entirely different. And in vaccines also, this genomics plays an important role now. So I just wish to talk these terms, and something like we have already heard about primordial, primary and secondary prevention in public health. Now it's the new term quaternary prevention, because we have to restrict the over medicalization. So in this aspect also, this genomics plays an important role. Yeah, that's.
Thanks, Dr. Naveen. So my next question will be to Dr. Unnikrishnan. So you have been an academician and then built a startup, exited. Currently, you work at one of the innovative centers in Kerala called CVJ Center of Synthetic Biology. So give us a glimpse about what synthetic biology is all about. We have a lot of young students here who can start a career in digital biology, synthetic biology. That's one. Second would be, what kind of industries can Kerala build realistically over the next 5 years, especially in these areas like biomanufacturing, synthetic biology? So what's the potential of these industries?
Thank you, Dr. Raju, Dr. Unnikrishnan, and the KDC team for giving me an opportunity to share my experience and opinion. And also, I congratulate Kerala government for initiating this particular initiative, KGDC, which is a very timely initiative to be taken, and it's maybe one of the first thing in the country. Coming to the question now, the synthetic biology. So, basically, when you talk about synthetic biology, you're talking about it's both two terms are coupled actually, basically synthetic biology and biomanufacturing. So here, what we do basically is there is naturally occurring certain pathways are already there in the natural biosystems. Like for example, certain bacteria, certain ethanol producing bacteria, certain specific pathways are there. Or in certain plants, terpenoid producing pathways are already there. So these are naturally occurring pathways. Now, when you understand a deeper genomic, through the deeper genomic approach on these pathways, basically, and can we, make use of this for synthesizing commercially important molecules? Maybe it can be a drug, or it can be, biofuel, maybe, food additives or industrially important chemicals. So what we do basically is you engineer this particular pathway, understanding the pathway, its enzymes, its cofactors, its intermediaries, and then engineering this into another biological system for the ease of manufacturing. For example, now a terpenoid pathway is there in the plant. Now, a particular terpenoid compound to produce from the plant, it may take maybe years to grow the plant and extract the extract. But now, if you engineer the whole pathway into a prokaryotic, system, or maybe eukaryotic system yeast, which has a turn around of maybe from a few hours to a day, and you get your product ready, basically. So basically, you're creating this pathway in a another organism, where allowing it to synthetically manufacture this particular compound, which is already produced in another natural biosystem. So this is like, synthetic biology is a prelude of biomanufacturing. Basically, you need to, using advanced genomic tools and now AI. We approach this problem and engineer the pathway into the system and then use it for biomanufacturing.
Dr. Shariga, you have been working in the science and technology space, and I know you are a policy researcher too, because you're trying to work on the new biotech policy. So, as you were interacting, the last biotech policy came some time back, and you're currently working on the next policy, right? So, what has changed the last policy, and how do you envisage creating a policy which also brings into our genomics and other allied fields like digital biology, synthetic biology? So where do you think it will reach in the next 5 years? Can we just avoid crosstalks in between, because there's a lot of echo coming up while we are speaking? So yeah, thanks.
Thank you, Dr. Raju, and the entire KDC for having given this opportunity. In fact, Kerala in biotech policy, it was initially formulated in the year 2003. with giving much thrust on to Kerala's biodiversity and the strengths that we had then, like agriculture, the traditional Ayurveda, then healthcare, education, etc., etc. So since then, there is, a whole lot of things have changed. Like, 2 years, 1 year back, we have seen one policy coming up, like the Bio E3 policy by the Government of India, the Department of Biotechnology, which is, seem to have giving more thrust to bio-manufacturing. So yes, we are currently in the process of revising this, the policy that we have initially formulated, and it is finally in the last draft stage for putting it in the public platforms and all. So here also we give more thrust on to human resource development. For the last panel, we have been discussing about the creating the skilling talent in diverse sectors, even in genomics. So we are currently in lack of premier set of people to be employed in the industry. So one of the main sector that we are going to focus is on the human resource development, and the second being the agriculture, which is one of the strengths of Kerala, and obviously the health sector, which we are in the foremost, and that too is giving a main trust upon. And also the emerging technologies like the artificial intelligence and machine learning, that is to be integrated into all these biotechnology platforms. So yes, so these are the main trust areas which is giving, which we are giving focus on in the new biotechnology policy. Obviously, that will be available in the public platform for everyone to give comments and inputs further also. So this is, yes, regarding genomics, we need to set up some infrastructure, like we don't need to have machines in every universities or every institute, but we need to have a centralized platforms where there will be infrastructure like the sequencers, the biobanks, everything in place in a centralized platform. And once that is being funded by the public sector, that can be accessible to all the industries and the institutions and whoever in requirement of it. And also we need to have a governance, like what was said earlier. We need to have a common data governance, like we need to have guidelines for accessing the data as well as the sharing of the data. So all these has to be into place. Thank you.
Yeah, thanks, Dr. Shariga. My next question will be to Dr. Naveen. So you have been trained in the US, and I used to jokingly say to you that you have the US CDC and now we have the Kerala CDC, right? And how to develop the next generation of skilling the professionals, because healthcare system in across the globe is doctors, you have nurses, you have allied healthcare professionals, including your workers, your ASHA workers, your pharmacists. So there are a lot of people working in tandem. And while the state devises a policy to make things happen, and now we have genomics, a lot of things are coming up. So how do you create the next generation of public health professionals to manage things properly? And Kerala being a very HDI state in terms of a lot of initiatives, but what can be done better?
It's a critical question. Even though Kerala is performing well in national level indicators, especially in Niti Aayog indicators, especially first in place, but we are just starting our journey in genomics. So we have to give proper capacity building to our health system because it's entirely new things, genomics, and most of the population are skeptical about the genetics, introduction of projects related to genetics and all, especially in Kerala. And many political conflicts are also there, especially when we touch genetic area. But in the post-pandemic era, especially after COVID-19, our public health system is very well equipped with many modern equipments, especially some of our public health institutions having NGS machines also. And in private sector also, they are equipping their labs with machines like NGS and all. And for example, in Calicut, one hospital have an NGS machine, and I tried to make it useful for making a genomic atlas for that institution. And I had discussion with the Dr. Vinos Karia and all. But having a machine is not a business, because developing a system for proper analyzing, reporting and all, it's very expensive. So a major hospital in Calicut, they couldn't do it. And especially they can make a lab and all, but they can give a proper report with proper, because I talked with many geneticists and they told me like they need a diagnosis which is precise. So making a new lab will be very difficult, and we should have a good governance mechanism which should coordinate the existing genetic labs together before starting new labs. And we should start from awareness campaigns, especially in population, and we should give proper training about the legal, ethical and social aspects of genomics to the healthcare professionals and all other stakeholders too. And we have to make a coordinated system. It may be a public-private partnership, because government alone can run this type of expensive things, and the importance of this good governance is the most important thing we should focus. And only after establishing the system, we should think about capacity building, because in the area of genomics, we are just starting, and we have to introduce all the aspects of genomics, its benefits, pros and cons and all. And some other things like DTC, direct to consumer, like reports, it should be handled very carefully, because it may cause more harms than benefits in the area of genomics. So these are the things I have to mention.
So Dr. Unikrishnan, my next question would be on being an academic and startup. What are the current gaps in academia and putting that into practice or entrepreneurship?
Yeah. So basically, academia is looking more at, at the, proving some of the mechanistic, or mechanism of the system. Whereas industry, the harsh truth is that basically who is the buyer and how much the buyer can pay for it. So this difference need to be understood to really translate what the technology to a commercial, purpose. But now what's happening is now, since we have incubators already available in many of the academic institutions, but owing to their presence, for example, now deep tech biotech processes like synthetic biology and biomanagement, which needs kind of more comprehensive infrastructure, which is not easily possible by an innovator to do it in the lab and simply using a proof of concept, just going to scale up, because scaling up is a major challenge in synthetic biology side or biomanufacturing side. Basically, if someone comes with you have already a proof of concept, someone is coming to you and asking you, say tomorrow, can you do a 10x scale? Because it needs optimization. So it needs more deeper or more comprehensive facilities, like I would say more like a biofoundry kind of system, which is very much essential to translate the deep tech kind of innovations. So that's definitely there is a gap in the stage. and I mean, in India, I mean, national level also. So that may be considered first. And of course, the market reality, because see, as a basic researcher, people don't understand what is the market reality. So that's very important to take in care of. So maybe, and I also feel that, many of these, the basic academic study grants, these are all decided mostly because if you look at the composition of the evaluation committee, they're more of academicians. But I would suggest if they can incorporate more, industry people into the system, or, commercial, like techpreneurs into the system, so that they can evaluate this and, see the weightage into the translation weightage of this particular project. That's very important, in fact. So that's one of the things.
Dr. Shariga, is there any initiatives from your department in terms of the translation research and commercialization part? How do we equip researchers to get into the patents being granted and also making it into a commercialization model? Because we see that KSUM has some initiatives.
Yes.
At the same time, Government of India is coming with a lot of initiatives. KDISC is also incentivizing researchers to go for patents and also commercialize it. So anything from your department that you're considering right now?
We have been so far funding basic research, giving more thrust onto that. But since few days, our priority has also changed. We are now encouraging translational research. In fact, at Kerala Biotechnology Commission, we have a program called the Industry-Linked Biotechnology Research Scheme, whereby we encourage projects which are of a problem of an industry to combine with an academics, and then a program of about 40 lakhs funding to be solved by the academics. That's a sort of an industrial problem. At the end of the day, that project which is being done, that has to end up with a commercializable product or process. So that is the mandate of that program. And apart from that, we are also in the process of creating incubation centers at almost all our R&D institutions under the Kerala State Council for Science, Technology and Environment, just to have this thing towards, converting what all happening in that R&D center towards commercialization. We are currently promoting, not much programs are focusing on that, but we are right now in the process of having such programs.
So any questions from the audience before we, yeah.
I just want to add that Government of Kerala has started a translational research and innovation center, the University of Kerala. It's almost ready, envisaging that we'll have startups, we'll have incubation centers, promoting translational research. Anyone who wants to translate their findings into a process or a product, I'm sure that this would be, because the infrastructure is ready, and I'm sure it's going to be inaugurated now. And this is one of the visionary programs of the Government of Kerala in Kerala University. And I'm sure that many of those translational programs, including the programs of Kerala Biotechnology Commission or others, can start work over there. Connected to that, we have CLIF, Central Laboratory for Instrumentation and Facilitation, where we have high-end instrumentation facility over there. So probably if anyone wants to have an incubation, to incubate their ideas, I'm sure this is going to be great. So that's a great initiative, Government of Kerala.
So I would like to add one point here. So this is, in fact, CBJ Center for Synthetic Biology Manufacturing is a very recently set up center. So in fact, we are now in a stage of scaling up the lab facilities. So what's happening now is, one of our thought is like, creating the lab in an already existing incubator system. So which is already in discussion with Kerala government, and it's already going to happen now. So that will, this will definitely, cradle the deep tech ecosystem in the state.
I would like to add just one thing. As part of Kerala CDC, in association with the CDAC, we are going to launch a new platform, Epiform. It's an epidemic forecasting and monitoring. So it's actually we have created disease models, especially for dengue, Nipah, lepto, and we are also working on hepatitis and all. So it's basically a data analytical tool with a dashboard system, and we have included many health determinants in this prediction and all. And in association with the KGDC, actually, if this genomic data is available, we can incorporate those data to this platform, and it will be launched soon. And the basic challenge is the difficulty to get a refined data. So our data capturing mechanism is also have to be improved a lot. So in view of capacity building and all, we should give proper training in areas of bioinformatics, data analysis and all. And especially we have posted nearly 100 epidemiologists in Kerala, and we should work together to prepare them for this data analysis and proper data capturing mechanism and all, and it will be a wonderful outcome. Yeah.
This is more of a technical question because you are discussing about synthetic biology. The configuration of the molecules are very important. Invariably that, nature is aligned for mainly L configuration. For example, if you see all amino acids are L amino acid. Now, when it comes some of this, let's say peptidomimetics or peptides, many times there is a tendency to add a D amino acid because of metabolic reasons. And many other molecules, when you are synthesizing, D configurations are there. So my question is that, is there any example the enzyme is engineered to make D configuration? I'm not asking whether you have done or not, but do you have any kind of example? I will tell you an example. Hydroxycitric acid is a D configuration which is naturally present, which is not normally there. And the enzyme is also mainly to make that one. You can't make synthetic D-hydroxycitric acid. That's why everybody is extracting from the natural this thing.
Yeah, thank you, Ramchand. In fact, enzyme engineering is also very commonly done in many of the prokaryotic platforms to, enhance, or, improve the pathway, this thing. Oh, specifically, I don't know, basically, about that. Because that's a very specific question. Probably someone who is really into, peptide bond synthesis may need to do. I don't know about that, yeah. But engineering, enzyme engineering is a very common thing in system, yeah. And to enhance the productivity or maybe, change the molecule, glycosylation or whatnot we can do. So, in fact, the non-coding DNA is also now, synthetically made. Peptides are being synthetically made and then, tried for their, potential for their therapeutic application, which is also another area where people are venturing into dark genome. That's another area.
Yeah, thanks everyone for the panel and being here for the Genomics Parliament. So now we break for lunch. Lunch is being served outside. We'll continue with the event for other programs of KDisc. The Honorable CM will be coming at 5:30 p.m. for the inauguration session. Yeah, thank you so much.